Sir, our Honorable Vice Chancellor sir has joined. Sujay sir, Pradeep sir.
Yeah. Good afternoon, sir.
Good afternoon.
Good
Yeah,
afternoon,
yeah.
sir.
Uh, Good afternoon to all of you, and thank you for joining at this webinar on computational biology and biomedical engineering. Your presence makes us very happy. I am Dr. Pradeep Sarkar, Associate Professor at the Department of Computer Science and Engineering, MAKAUT, the program coordinator of this webinar. On behalf of CSE Department, MAKAUT, it is my privilege and pleasure to welcome you all. I wish you all a cheerful welcome. Also, I wish a convivial welcome to the eminent speaker come from world-renowned organization of India and abroad to share their knowledge and enhance our mental horizon. Today's webinar has about 200 participants from different parts of our country and abroad, making this webinar a grand success. Since the last 1990s, the computational biology has become an important part of developing emerging technology for the field of biology. Computational biology and biomedical engineering is a rapidly developing multidisciplinary domain where procedures and methods from computer science are developed and deployed to address, to analyze large collection of biological data, such as genetic sequence, cell populations, or protein samples, and create accurate modes of the human brain, and also solve important current problem in biology. The computational methods used, analytical methods, mathematical modeling, and simulation, and machine learning. The aim of this webinar is to present the latest research and result of scientists, researchers related to computational biology and biomedical engineering. This webinar provides opportunities for the different areas, delegates to exchange new ideas and application experiences to establish research mission and to find global partners for future collaboration. We hope that the webinar results constituted significant contribution to the knowledge in the scientific field. Now, we are delighted and proud to announce that our Honorable Vice Chancellor Sir, Professor Dr. Shoykat Moitro is present here and will deliver his inspirational speech and inaugurate this webinar. A dream does not become reality to magic. It takes sweat, determination, and hard work. Yes, it's a belief endorsed by our Honorable VC Sir. He has always been our source of inspiration and motivation. We'd like to express our most sincere gratitude for your presence in this opening ceremony as the gateway to the initiation of this webinar. So welcome our respected VC Sir. Please, sir, inaugurate this webinar.
Thank you, Dr. Pradeep Sarkar and Dr. Sujoy Mistry for organizing this webinar, combining two different aspects. One is soft part of this biological researches, computational parts. Another is biomedical engineering. Perhaps this different designing part of it will be discussed. Now, there is a great thing that you are organizing this very important topic for the this benefit of all the participants here. A few days before, we had other programs of computational biology, and the program in biomedical engineering is happening for the first time. So I think we have some more experts with us in biomedical engineering. To kindly invite Dr. Shivamay Dasgupta, because he has some knowledge and experiences in biomedical engineering. So he may be one of the important, this panelist member for the panel, either for coordination or moderation or for sharing his views. Now, regarding this importance of computational biology, is basically this area is growing. This area is as an interdisciplinary field of research. It is an emerging field. It is growing. It combining biology, computer science, information engineering, mathematics, statistics, for analyzing, for interpreting, for gaining meaningful insight from the biological data. And this because this now this data is going to be a very this crucial and critical assets for each and every activities and each and every these organizations in biological science also, particularly in Indian context. We are generating a huge volume of data. Now earlier we did not have much scope is handling this huge data, but now but at this present moment, because of this phenomenal growth in information technology and this emergence of subjects like big data analytics and all these things, this is now quite formidable thing. So basically, understanding of different things, biomolecules or DNA sequencing or evolutionary genetics or drug development or precision medicine, this data analysis, data interpretation, by deploying many tools and techniques. This is now quite this formidable, and based on this, the subject is getting a momentum, and lots of these mathematical tools, techniques, programming languages are coming up in facilitating that process. And global market in this area is also increasing in the field of computational biology, bioinformatics and related areas. This global market is increasing rapidly, and there will be these large numbers of this scope for employment and for engagement of our present this researcher, present generation of students. So now maybe in near future we may introduce academic programs in computational biology as well, apart from bioinformatics, because this in the coming decades, by 2025, 24, 25, we will be requiring numbers of professionals in this field, in the in computational biology, health science, genetics, genome, drug discovery, molecular biology. There's a huge scope for life science industry professional is coming up. So this is very important. Biomedical engineering is also important in that sense. Now understanding these bio-inspired processes, now this biomimicry and designing things. This also combines this many important the understanding of robotics, understandings of these different types of these electronic gadgets and items. So this is also very important, particularly in this country. We have a huge this market for all these things, and this is emerging right now. There is not much this awareness in computational biology, biomedical engineering, by and large in the entire academic community in scientific field. But as a potential area, its scope and applications in near future will be enormous. Therefore, these types of programs which you have organized is of quite importance, because it will create awareness. It will create this level of understanding, basic level of understanding among all the learners. And out of this, many of these participants or learners, they might be interested in pursuing their researches and pursuing further the studies in this area, and that will create a knowledge community. That is our expectation and our one of the mandate. And for doing that, these types of these programs in this line, it needs to be organized sequentially at regular interval, inviting all other experts as well from as you have invited these times from different corners of this world. Now, this is one of the advantage of this present time, that assembling experts from different institutes, different universities, at these geographically located at different parts of this world. Now it's quite easy because of this digital platform. And otherwise, in the normal time, it would have not been that much easier. But right now, with the shortest possible time, you can organize these types of training programs. And my thanks to my sincerest thanks to all these resource persons who have kindly consented to be amongst us for sharing their knowledges, for sharing their experiences and ideas and opinions for the benefit of our participants. And I believe that in future also they will be available to take up these types of training programs at higher levels for the benefit of our existing participants and also new participants. So with this, I'd like to thanks to Dr. Mistry and Dr. Poddot Sarkar again, and my sincerest thanks to all the organizing team of Computer Science Department. And I wish a very effective and and beneficial learning and useful learning for all the participants present over here. Thank you all.
Thank you, sir, for your motivational and informative speech. And we always need your support and motivation. Shall I start the program, sir?
Yes, yes. Kindly start this program.
Okay, now the flags are flying, the balloons are already released. It's a great one we have been planning and waiting for. Now, welcome Dr. Sujoy Mistry, the program coordinator of this program and assistant professor of CSE Department MAKAUT. Please host the first session of this webinar. So welcome Dr. Sujoy Mistry.
Thank you, Dr. Poddot Sarkar. Thank you, sir, Honorable Vice Chancellor, sir. Thank you all. Good evening to all. This is a proud moment for me, and especially proud moment for me for that I am now going to host one of the eminent person of this field, that is Dr. Subhashish Barik. Dr. Subhashish Barik has completed his bachelor's and master in microbiology from University of Calcutta. He completed his doctoral study from Chittaranjan National Cancer Institute, Department of Life Science and Biotechnology, Jadavpur University. In doctoral research, he studied cancer immunology, specifically how modulation of tumor microenvironment can be strategy to restrict tumor growth. Soon after completing PhD, he moved to the Department of Molecular Biology and Immunology, University of Missouri, Columbia, USA, for postdoctoral research. He did his postdoctoral training under the eminent immunologist, Professor Dr. Habib Jahoni, where he studied T-cell development and autoimmunity, neonatal immunity, etc. He received his DVD Ramalingaswamy Fellowship, which is considered one of the most prestigious fellowships offered by Department of Biotechnology, Government of India. Presently, he is holding permanent scientific position in the Department of In Vitro Carcinogenesis and Cellular Chemotherapy, Chittaranjan National Cancer Institute, Kolkata. He has published more than 28 different peer-reviewed international journals. So now I am welcoming Dr. Subhashish Barik to delivering his enlightened speech. Dr. Subhashish Barik.
Thank you, Sujoy. So first of all, I would like to thank the organization committee, the Honorable Vice Chancellor of MAKAUT, and especially Dr. Sujoy Mistry for inviting me and giving me opportunity to speak over here. So I'm sharing my slide. Taking little bit time. So shall I start?
Yeah.
Yes, yes.
Yeah, okay. So my today's topic is T cell development and leukemia, a symbiosis between wet lab and dry lab. So here I am going to speak why T cell development is important to explain the T cell leukemia and why it is important to make a collaboration between wet lab and dry lab. So why T cells? T cells are a type of blood cells which comes within the white blood cells called which are called WBC, and they are known to prevent infection. The major jobs of T cell are to fight specifically against pathogen, and it has a power to discriminate between self antigen and foreign antigen. And for this job, they have extensive training at different schools called lymph node or thymus. In the thymus, they are undergoing two kinds of selection, positive selection and the negative selection. In the negative selection, all self-reactive T cells are deleted. And after completion of their job, some of them are preserved as a memory cell, and that's the reason they are very important for adaptive immunity and vaccine development. Now, what happens if we delete T cells from an individual? If you look at the picture, the white mice is a normal mice. It is FOXN1 sufficient. This is a wild type mouse. It has a thymus and T cells, everything, entire immune system. But if you delete the gene FOXN1 from this mice, it will become the nude mice. It lacks the thymus and it does not have uh, T cells. So these mice are susceptible for infection and susceptible for tumor development. Now, if you now it is known to everyone the cancer develops from the normal cells. But unlike other cancers, T cell leukemia is something different because it develops from the progenitor cells. In case of T cell leukemia, two different kinds of progenitors are there. One is multipotent T cell progenitor from where ETP-ALL develops, and another is unipotent T cell progenitors from where TALL or TALL develops. Now, ETP-ALL is early T cell progenitor acute lymphoblastic leukemia. It is basically a small subset of TLL. It is multipotent in nature and its lineage fate has not been decided. So they are multipotent. Now, before going to say something, I must go through the process hematopoiesis, not just to refresh your knowledge. In order to differentiate into different cell types, hematopoietic stem cell has to divide in two different subsets. One is common lymphoid progenitor, that is called CLP, and another is common myeloid progenitor called CMP. CMP gives rise to lymphoid cells like B cells, T cells, NK cells, etc. Whereas CMP gives rise to different uh, macrophage or dendritic cell like APCs and other granulocytes. And CLP or common lymphoid progenitors, they never ever give rise to myeloid cells, and these myeloid progenitors CMP, they never give rise to B cell, T cell, NK cell like lymphoid cells. And this is called the basic principle of the hematopoiesis. Now, if you focus on the T cell development, the bone marrow derived stem cells, those who settle in the thymus and give rise to T cell, are called early thymic progenitor cells or early T cell progenitors. Previously, it was conceived that they only give rise to T cells, but later on, it is evident that they can give rise to lymphoid like B and T cells, as well as myeloid like macrophages and dendritic cells. In fact, these early thymic T cells or early thymic progenitors, they are major granulocyte precursors in the adult mice thymus. Now, in order to develop T cells, these stem cells has to undergo different stages. And here is a fax plot, and each dot represents a single cells, and they are separated based on c-KIT and CD25 expression. CD25 indicates the T T cell way. I mean, they are going to the T cells. So see here, here is DN1 ATP, and the next step is DN2A, and then DN2B, then DN3. Then it becomes to CD4 positive, CD8 positive cells. So here DN means double negative. They are CD4 negative and CD4 positive, CD8 positive. And after DN3, there becomes double positive cells, that is CD4 positive, CD8 positive. Then it undergoes two types of selection. One is positive selection that gives in the survival license, and then it has to undergo under negative selection, where the possibilities of all self-reactive T cells are negated. Then it comes into medulla of the thymus and then get out from the thymus as a recent thymic emigrant as a CD4 positive T cells and CD8 positive T cells. Now, if you again focus the T cell development from the hematopoietic stem cell, you will see in order to make the 100% commitment, it has to pass different stages. And interestingly, in every stage, it is losing some characters or some traits that help them to become other cell types. So we question that CLP derived ATPs, which has to go to the T cells, why they do not follow the conventional T cell route, the developmental route, rather the choice into the myeloid differentiation, why they do turn off the why they do turn off the T cell developmental program and turn on the myeloid program, what causes them to give up the multipotency, and what instruct them to break the basic principle of hematopoiesis was really unknown. So in this area, our lab, I mean our postdoc lab, was the first lab to say something. They said the ETP are not the homogeneous, rather they are heterogeneous and they can be subdivided based on IL-13R alpha 1 receptor. And they found that the IL-13R alpha receptor positive cells, they go to myeloid cells and they do not go to T cells, whereas the negative cells, they go to the T cells. Before going to other things, I should describe what is IL-13R alpha 1. So IL-13, the cytokine has two kinds of receptor. One is IL-13R alpha 1 and another is IL-13R alpha 2. IL-13R alpha 2 is a decoy receptor. It can bind with IL-13 only and cannot transmit the signal, whereas IL-13R alpha 1 can complex with IL-4R alpha and can bind with both IL-4 and IL-13 and transmit the signal. That's why it is called the heteroreceptor, and from now I will call it heteroreceptor or HR. In order to test the role of IL-13R alpha 1 in this regard, we use three different tools. One is IL-13R alpha 1 or HR deficient mouse. This is the knockout mouse. And next thing is IL-13R alpha 1 GFP reporter mouse. In this entire mouse, the cell is expressing IL-13R alpha 1. They become green or GFP positive. And the last one is anti-IL-13R alpha 1 monoclonal antibody. Then we use two wonderful systems, that is the OP9 cell line and OP9-DL1 cell line. The specialty of OP9 cell line is it helps in the myeloid differentiation, that is macrophage or BC differentiation, whereas the OP9-DL1 cells has DL1 receptor and they promote T cell differentiation. So if you use these two kind of cell lines, you can culture them, or you can make the T cell development within a petri dish. So previously we know that HR positive ETPs, they do not go to T cells, as the cultured on OP9-DL1 cells for the 3 days. So we question that, is it a permanent defect or it is due to a slower kinetics of T cell development? In order to test that, we took the thymus, then we isolated the ETP, we sorted them as HR positive and negative, and then cultured an OP9-DL1 cells for different days, 3 days, 7 days, and 10 days. And up to 10 days, we found there is no T cell when you use HR positive ETPs. whereas the negative one is giving the almost 100%. So HR positive ETPs, they do not go to the T cells. But when we culture the both subset on the OP9 cells that is supposed to help in the myeloid differentiation, we found both of them are giving the myeloid cells, including the dendritic cells. Again, it is again the IL-7R alpha. This is very important receptor for the survival of the progenitor T cells. So, and FLT3L, FLT3L, it is a inducer of the IL-7 R alpha. So we treated FLT3L in the HR positive ETP. We found still it is not expressing IL-7R alpha. So we concluded here that these guys, these HR positive ETPs, they are so adamant. They don't want to become the T cell, even if we push them toward the T cell lineage. So we concluded HR positive ETPs are fixed. They can only go to the myeloid lineage, whereas the negative ETPs, they are flexible. They can go either with the myeloid or lymphoid lineage, whatever the environment we can provide to them. So then we questioned that does HR inhibit the lymphoid development? In order to test that, we use a cloning of IL-13R alpha 1 in a MSCB 1 Thai 1.1 vector, and then it is transduced retrovirally into HR deficient ETPs. It was isolated from HR deficient mouse because they haven't seen the HR signaling before, and after 3 days we sorted HR positive ETPs based on Thai 1.1 positive, HR positive using the antibody. Then we cultured on OP9 DL1 cells for 7 days, and we found when we treated them IL-4 and IL-13, the T cell development was markedly reduced. This indicates that IL-13R alpha 1 inhibits lymphoid lineage specific development. But in order to be sure that IL-13R alpha 1 or HR controls the lineage commitment, we developed the HR positive ETP from the same type. That is, we took HR deficient ETPs from knockout mice and generated HR positive ETPs. And then we took a 96 well plate preceded with OP9 DL1 cells, then sorted a single cell into the single well. So each cell contains only one cell only. Then we cultured them with IL-7 and FLT3L in presence or absence of IL-4 and IL-13. And in the result, we found that when we added the IL-4 here or IL-13, the T cell potential was decreased 100% to 18% and 100% to 13%. It indicates that IL-4 and IL-13 directly inhibit the T cell lineage potential of ETPs. And in other experiment, we found that in HR positive ETPs, they express IL-7R alpha very low, and they do not express the notch one, whereas the negative one, they express high on both of them. Now, in order to become a T cells, a progenitor cells must express notch one, which should interact with delta 1 with OP9 DL1 or some epithelial cells of thymus. Then it expresses the TCF1, a transcription factor, which gives rise to different transcription factor and other things that develop the T cell development. So we concluded here HR positive ETP display minimal expression of IL-7R alpha and Notch1 receptor. Now, if you look at the progenitor cells, there are two different transcription factors. The T cell progenitors. One is CEBP alpha, which is a trademark for myeloid lineage, and another is TCF1. As the name implies, T cell factor 1, it drives the T cell lineage commitment. And in this entire picture, these two transcription factors, they are basically competing. If TCF1 win, it will go to the T cells, and if CBP alpha win, there will be a myeloid trait. But in this picture, there is another protein called HAZE1, which can physically inhibit the CEBP alpha and promote the lymphoid development. And the interesting thing is that both transcription factor Hayes1 and TCF1 is under notch and control of the notch. So we tested the Hayes1, TCF1 and CEP alpha. We found the positive one has less Hayes1, less TCF1, but more CEP alpha, and negative one has a higher Hayes1, higher TCF1 and CEP alpha. So we concluded HR positive ETP contained a decreased abundance of factors necessary for the T cell development. And we also looked at different transcription factors and other things under control of TCF1, which is under notch 1. We found in case of HR positive ETPs, that the white box, they are expressing very less, whereas the negative one, they're expressing it's a lot. So we concluded inhibition of notch 1 must be the crucial step in the lymphoid lineage specific gene expression in HR positive ETPs. Now, so we hypothesized here HR signaling somehow inhibiting notch 1 and thereby inhibiting T cell development. If you look at the notch 1 transcription, notch 1 itself helps its own transcription. That's in a positive feedback loop with the help of E2A, another transcription factor. Now here there is another protein is called IB3, which can sequester this protein E2A out of the nucleus and can inhibit the notch1 expression. Again, we looked at the signaling of the IL-4 and IL-13 through heteroreceptor. It is in the literature that it can activate STAT1, STAT3, and STAT6. So we look at the phosphostat6, phosphostat1, and this like this. And we found only phosphostat6, Y461, and phosphostat1, Y701 is upregulated. So in the HR positive ETP, whatever is happening is through these two. Now from the other experiment, we found that IL-4, IL-13 can interact with this heteroreceptor, which can activate phosphostat1 and phosphostat6. By activating phosphostat6, it is induces ERK1, which basically activates EGR1, which induces the expression of ID3 and thereby reduces the expression of notch1, and that's the reason HR positive ETPs do not go to the T cell. We tested this in vivo with STAT6 inhibitor. So we took IL-13 R alpha 1 GFP reporter mouse. We injected STAT6 inhibitor and then we looked at STAT6, PERK1 and ID3. We found when we use this inhibitor, this purple one, the level of phosphostat3, STAT6, ERK 1/2 and ID3 was decreased. And we did the same experiment in vitro using the retroviral transcription factors. So we found that this is the reason they do not want to become the T cells. We did another in vivo experiment. We used the same mice and we treated them with STAT6 inhibitor and ERK 1/2 inhibitor. And then we isolated HR positive ETPs and looked at the notch1 expression at the mRNA level. And we found that when we are using inhibitor, whether it is STAT6 and ERK1K2, the notch1 expression is back to the normal almost. So if we can inhibit the STAT6 or ERK1/2, the cells can become the T cell. In order to test it, we injected STAT6 inhibitor within the same mice, isolated HR positive ETPs, and culture on the OP9 DL1 cells. And we found there is at least some T cells are there. This is plotted against the CD11B versus CD25. So CD11B is a marker of the myeloid cells. It is not going to myeloid, and it is cultured on OP9 DL1 cells, but it is going to T cell direction. And we also cross-checked with another cloning, that is TCF1RB. We transfected HR positive ETPs with TCF1 in just to overexpress TCF1 over there. And then we isolated the TCF1 overexpressing HR positive ETPs and culture and one dopamine DL1 cells. And we found that upon TCF1 overexpression in HR positive ETPs, the cells are going to the T cell direction. So HR signaling inhibits the T cell development in the ETPs. We also tested several transcription factors like RELB, N50, ZBB46, IRF8, and so on, which are mainly for the disease up to here, and this is PU.1 for macrophages and B cells. We found HR positive ETPs are expressing them a lot other than the B cells one. And then we did another experiment. We used a STAT1 and STAT6 inhibitor in the in the IL-13 or alpha-1 GFP reporter mouse, and we isolated HR positive ETPs and looked at the DC transcription factor. We found, interestingly found, the only IRF8 is regulating, it's regulating under the STAT6. This is the black one. So when we untreated, they are showing a lot of IRF8. When you use the STAT6 inhibitor, they are showing a lot of IRF8. But when we use the STAT1 inhibitor, you can see it is go back to the baseline almost. So we concluded that HR signaling induces IRF8 expression to STAT1 activation, and HR-driven STAT1 and STAT6 activation is independent to each other. In order to test whether the HR is the important molecule to become dendritic cells and don't want to become the T cells, we use the experiment using congenital mice. So we use the HR deficient mouse and isolated HR deficient ETPs, and then overexpress HR with them HRIP, and we isolated HR positive ETP from there, and then injected intrathymusically into CD45.1 congenital mouse. And after 15 or after 2 to 3 weeks, we isolate the thymus and leveled with the two kinds of antibodies, CD45.1 and CD45.2. And then we gated on the CD45.2 because we took this cell, which was CD45.2, and we are basically this is a barcoding system, and using this barcode we can trace that. And we looked at the T cell, we found still they are not expressing the T cells, a T cell phenotype which is a CD3. So when we can express them with HR, it is not going to T cells. And when we looked at the myeloid population, we found a lot of them are going to the CD11P positive, CD11C positive disease, and they are mostly CD8 positive. So HR helps them to become CD8 positive dendritic cells. In another experiment, we show that if you use the STAT6 inhibitor, the percentage of the CD8 positive dendritic cells is getting low. So we concluded that the HR can interact both with IL-13 or IL-4. Using IL-4, it can activate the activate the STAT1, which can induce IRF8, a transcription factor that drives CD8 alpha positive CDC development. And in the other way, it can use the STAT6 activation, which ultimately induces the transcription factor ID3, and it inhibits the Notch1 expression and thereby inhibit T cell development. In the other experiment we found HR positive ETPs contribute to intrathymic resident DC pool. So in the summary of this work, we can say HR can be used as a marker for the linear fit choice. It can become a myeloid, it can become a lymphoid cells into the myeloid cells, and it make it the dendritic cells, which basically helps in the negative selection and helps in the protection from the EAE, which is experimental autoimmune allergic diseases, encephalomyelitis, which is basically a neuro disease similar to multiple sclerosis. So we concluded that the thymic cytokine environment regulates the central tolerance by alteration of the lineage commitment in T cell progenitor cells. Now, if we back to the leukemia, mainly T cell leukemia, if they arise from multipotent T cell progenitor like ETP, it can become to early T cell progenitors acute lymphoblastic leukemia, that is called ETP-ALL. And if it originates from the unipotent T cell progenitor like DN2B or onwards, it can become to acute T cell lymphoblastic leukemia like TALL. Now, the ETP-ALL, they are basically a small subset of the TALL. It corresponds 11% of the childhood and 7.4% of the adult T cell acute lymphoblastic leukemia. It originates from ETP and characterized by a very early differentiation arrest and show unique genetic and transcriptional features. They are considered as high risk prone and very due to its very poor prognosis and worse outcome. It is multipotent and its lineage fate has not been decided yet. So here our plan is to work whether microenvironmental factors can control the robust proliferation of ETP leukemic blast by targeting their multipotent nature, and whether they can overcome the effect of genetic mutation in ETP-ALL patient in East Indian population. Basically, we are looking for the looking to target the multipotent function. If we can change their mind from lymphoid to myeloid, automatically it will lose its robust proliferation potential, and that can help the patient to get the at least a good quality of life. In order to study, we use the different models, and one is the murine ETP, because ETP-ALL have the lots of similarity with murine early T cell progenitors. Now, if you look at the T cell development from the HSCs, goes to LMPP, then CLP, then ETP or DN1 subset, then it goes to DN2A. And this single transition, it takes 10 cell division. So here question is why the developmental process is so slow, and there must have several checkpoints that restrict them to become the T cells. And our plan is to work that we will look at the each cell division, and we'll study them at the transcriptional and epigenetical level to explore their role in stepwise commitment in the T cell lineage, and whether the ETPs, they use the receptor and other than T cell, other than HR, to escape the T cell development within the thymus. In order to address these issues, we did some in silico analysis. And before going to tell this, I want to say that I basically illiterate in this field. I'm trying to understand it, and my students are also helping a lot to understand this. And here we took some NCBI geo database, and we took a database that has the 6 population HSCs, LMPP, CLP, ETP, DN2, and DN3. And we looked at the pan cytokine receptor expression, and we make a heat map. And from the result, we found the LMPP and CLP, they are more closer than the HSC, and same trend was found when we looked at DN2 and DN3, they are more closer than the ETP. And from this heat map, we prepared 2 panels. One, here is the different cytokine receptor, and we tabulated it here because this receptor expression is upregulated from ETP to DN3 stage, and the lower part here the receptor was steadily downregulated from ETP to DN3 stage. And then I want to show this one because here is the ETP, which is multipotent, and then is the DN2A, it is also multipotent. So at least here they have some T cell, they have some myeloid ability. They can still go to the myeloid lineage. So there is a commitment between DN2A and DN2B. So T cell lineage commitment occurs here. And then after DN3A, they express TCR beta and makes a beta selection. So when it is under DN2A and DN2B, it can go to the T cells, NK cells, gamma delta T cells like this. But once they undergo the beta selection procedure, then it is definitely CD4 or CD8 positive T cells. So we took another NCBI geodatabase where we have the ETP, DN2A, DN2B, DN3A, 3B, and DN4, and we did the same heat map analysis, and we found, and in order to look at their variance spatial distribution or their distance between them, and we found that DN4 and DN3B, they are more close. If you look at the heat map, it looks the same. They look same. And if you look at the ETP and DN2A, they are not similar basically. They have a lot of distance, and if you look at the DN2A and DN2B, the distance is less, and if you look at the DN3, the distance is more or less. Now, what is the consequence of this receptor signaling? In order to test that, we used some transcription factor importance for the survival, their development, and everything, and using the same geodata sets. We used a k-mean clustering, three-dimensional scatter plot constructed using Excel stat, and we found 4 different clusters. Here we have 3 axes. X-axis denotes the transition of ETP to DN2A, Y-axis DN2A to DN2B, and Z-axis shows DN2B to DN3A. And we have 4 different clusters: the green, blue, red, and yellow. Green clusters contains the receptors like EPOR, IL2RG, IL9R, and like so on, and it has the transcription factor like beta T, the Annex1, TCF1, TCF7, TCF12, and they are probably involved in launching the transcriptional program initiating T cell development. Whereas the blue cluster, it has the other receptors, and the transcription factors related to these are here, and they are probably involved in inducing myeloid fate and regulating the proliferation of in progenitors. The red one helps in the involvement in restricting the progenitor potency towards the T cell lineage, and the yellow one probably involved in maintaining the flux of signals toward the T cell lineage developmental program. And in from the other papers, if you look at the ETP-ALL, Zhang et al. Described 3 different classes of genes, like activated cytokine receptor and RAS signaling genes related to hematopoietic development, and histone modifying genes, which are important in the ETP-ALL. So we isolated mouse thymus and sorted these populations from here, and harvested and isolated the total RNA and did the real-time PCR analysis, and we found the several plots here. Some of them are decreasing and some of them are increasing in the ETP to DN4 axis. And if you look at the cumulative data, that we found KRAS, FLT3L, JAK3, SH2B3, and ETP sites, and they are decreasing, and they may be involved in the T lineage commitment, whereas the genes associated with the robust proliferation, we suspect that some of them are down regulating. It is so the first one is down regulating from from ETP to DN2A, and it was down regulating from DN3. I mean here from DN3, DN2B to DN3A. So they can be important as a gene associated with robust proliferation potential, and this indicates that last 2 the GATA2 and DNM2, they are directly associated with robust proliferation potential in a positive sense, whether they are important or they may work as a negative sense. So we need to do some wet lab validation, and this is also a wet lab data. Still, we know we wanted to know the role of these genes in the T cell development, whether it is murine or human T cell development, which we want to do from the cord blood, human cord blood, CD34 positive cells. And here is my publications, and here is my collaborators, Dr. Habib Jagwani from University of Missouri, Dr. Ganguly and Paul from IICBCSR Kolkata, Dr. Chakrapani from Apollo Clinicals, and Dr. Dr. Chaudhary, Dr. Mukherjee, and Dr. Alam from the CNCI where I am. And thank you for your patience. Thank you all. Hello.
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 Hello, 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 hello. Hi, hello. Thank you, Dr. Subhashish Barik. Thank you, Dr. Subhashish Barik, for your such knowledgeable and enlightened uh, slides and talk. So now we can maybe take one or two questions maximum. After that, if anyone have any query, then at the end of the session, at the end of the this webinar, we can share the eminent speakers mail IDs. You can if you have any query, then you can directly mail him, and you can clear your doubt actually. So till now, I have just got one question from Ovik Mitro. That is, sir, do you have what type of mathematical model you are using or statistical model you are using for analyzing those data, whatever you're saying? That only question has still now arise.
Okay. For this clustering, we use Excel stat, and still I do not understand it properly. Then we need to validate we need to contact with some bioinformatician. We have a collaborator, and we need to recheck this one. So basically, I am.
Okay.
We use most of the online software.
So if anyone have any questions, otherwise I will share Sar's mail ID in the later section. Okay. Thank you, Dr. Subhashish Barik. Okay. If you any questions in the end of the webinar, I will share all the information how we communicate with the sir. Then you can, if you have any question, you can clear your doubt through his mail ID. Okay. Thank you, Subhashish Barik, for your talk. And I will pass first to Dr. Poddu Sarkar to continue this session.
Thank you, Dr. Subhashish Barik, for your nice presentation. And now, the second eminent speaker, Dr. Ahmed Ibrahim Shamir Khalil from Singapore. Ahmed Khalil is a postdoctoral research fellow at the School of Computer Science and Engineering, NTU Singapore. He received the PhD degree from NTU in 2020. He got his BSc degree in 2011 and MSc degree in 2015 in Electronics and Electrical Communication Engineering from Cairo University in Egypt. In early 2021, he will join Harry Perkins Institute of Medical Research to study the tumor evolution and ecosystems using single cell model omics data. Dr. Khalil's research interests include a broad range of topics around computational biology, data science, and hardware acceleration. The title of his talk, Copy Number Correction, an accurate interpretation of chromatic conformation of cancer cell lines. So heartily welcome Dr. Ahmed Ibrahim Shamir Khalil to present his paper. Thank you and welcome Dr. Khalil.
Okay, can I start?
Yeah.
Okay, thank you, Dr. Sarkar, for the invitation and for this great introduction. and good evening everyone in India side, and good night everyone on Singapore side because the time difference. And today I will talk about mainly about my work during my PhD, which was about like accurate modeling of cancer cell line. And at the end, I will make like overview of my future work or my current work for single cell data. So let me share my screen. Okay. Yeah. So now it is clear on my screen.
Yeah, it is visible. Okay, it is visible.
Okay. So mainly today I will talk about my work during my PhD that I finished in 2020. So the title, as we see, that it is copy number correction for accurate interpretation of chromatin conformation of cancer cell line, which we can say it is a big title, but we can divide this into four main terms, which is copy number correction and chromatin conformation, and using this data to accurate interpretation of cancer cell line. So in general, what our approach or what our objective is usually to analyze or cancer cell, and to do the objective is to do accurate modeling using kind of data integration. So if we give like some introduction or quick motivation, that as we all know that cancer is one of the most leading cause of disease. In 2018, it is resulted in 9.6 million cancer disease. And as we all know that cancer research aims to develop methods to detect, diagnose, and treat cancer disease. However, that it is not on all cancer types, there is some still poor clinical outcome. One, in during my PhD, it is we will focus in human cancer driven cell line because it is the main experimental model for cancer research, and it is used for developing and testing drug therapy. We found that accurate modeling of this cancer cell driven cell line are still challenging because two main points. One is the widespread and dynamic change of genomic alteration, and second is tumor heterogeneity. And based on this, we in my PhD we focus about developing computational solution for accurate interpretation of 3D genome confirmation by integrating the copy number variation. So our main idea is how to do accurate interpretation of 3D genome confirmation of cancer cell line using the information of copy number variation. And for this, we developed two computational solutions, one in Sinatra and High Sinatra. So this was the work during my PhD. And at the end, I will talk about single cell multi-omic data integration because this is the current hot topic or like emergency technology, we can say. So let me first start by quick introduction of copy number variation. As we all know that copy number alterations or genomic alteration are hallmark of cancer cell. So if we talk about normal cell, that we expect that we have two chromosomes, but in cancer cell, we will have different variety of this chromosome. We may have one additional chromosome. This is called chromosomal duplication. One less deleted chromosome, chromosomal deletion, and we may also have like increment region like duplication or amplification, like here in H1 region, it is duplicated here, or removal of this region, which is called segmental deletion. And each cancer cell, or especially cancer cell line, have combinational of this genomic alteration. Extra addition to this, that cancer cell line or cancer cell go under adaptive process to gain more copy number event. So if, for example, we have this initial cells, we may have some kind of copy number alterations or genomic alteration, like H1 amplification, chromosome 2, one extra copy of chromosome 2. But after saving this after passage time, we may have more event like here the amplification of F3 region of chromosome 2, like we have chromosomal gain or segmental gain or segmental loss. So based on this two, for accurate modeling of cancer cell line, we need to identify the copy number information from the same data we have, because we may, for example, many cell lines are used by different labs, but we may have some kind of clonal variability as the difference between the same cell line from different labs. For example, MCF7 breast cancer cell line and so on. So what we did, we looked at about how to detect this copy number variation. And the most common way to detect the copy number variation is to utilize the RD signal. So quickly, the RD signal is represent the count of mapped sequence read at each reference, each location of the reference genome or each loci of the reference genome. So if we have any GS read, we map from any experiment, we can map this to the reference genome like HG19 or HG18. And based on the data, we can model this data to identify loss or the deletion of some region, like what we call segmental deletion, or here's the gain, which is called segmental amplification. This is the common approach people use for detecting copy number. But to understand this more, let's compare between one normal individual like HG00119 cell line from 1000 genome project and compare the RD signal of this data to the cancer cell line. So for this, for example, we can make the main feature of the data, like most of the chromosome have similar number of reads as we see here. And even if we make zoom in one chromosome, we'll find we can consider this as a normal distribution, for example, and we can detect the amplified region or the deleted region, and this is the centromere, for example. And for this, we will have the frequency distribution represented by unimodal distribution, like here. And based on this, most of the copy number detection tool model the RD signal using unimodal distribution, like Gaussian, Poisson, negative binomial distribution. But let's compare this HG00119 cell line to the A427 cancer cell line. We will find huge difference. So if we look here, first, the RD signal between the chromosome is not similar. As we see, we have different level of or different counts, which represent the number of reads in each region. And if we make zoom here, same, we will find that feature of cancer cell line, we have large structure variation here. It is not smooth like here. And if we make zoom, we will find also some case of wave artifact. And as out of this, if we make the frequency distribution of this signal, we find that this represented multimodal distribution, not more same like unimodal distribution here. So for this, what we did, as a feature of cancer cell line, we can extract, we have multimodality of the RD frequency distribution compared to unimodal here. And we have widespread large scale copy number variation, which can result from chromosomal segregation here. And plus to this, we have many focal alteration, which people usually call about copy number variation or small copy number variation, which we believe that this is more correlated or linked to gene expression. So if for example, this one gene can be expressed, we expect this is expressed because here amplification of this signal or repeat of this signal in the RD signal or the DNA sequence. So the main question with us is how to model this RD signal from the same data. We don't need to depend on previous data. And so what we did here, we analyzed more cancer cell line. This is the only normal here, which we can find that estimated copy number of the 2n which means that most all the genome have the copy number of 2. We have 2 copies of each region of the chromosome of the genome. But if we go for all cancer cell line, we have different combination. Here, for example, in IMR32, we will find that the majority is 2, but there are some region which is amplified and some region deleted. Same HB212. But for example, if we check MCF7 breast cancer cell line, we'll find that the the majority have copy number of 3 and 4 and 5. So how to model this all from the same data? This was the main question, because we are targeting to accurate interpretation of the 3D genome conformation. So what quickly 3D genome conformation? 3D genome conformation, it is mainly that two main approach is high C and 3C set that developed to capture the special 3D genome conformation by identifying the chromatin contact between genomic regions. So this is usually, it's targeted to find that the revealing how the gene how the gene is activated. For example, if we look here, we all know that some kind of gene activation is transcription factor come to bind in the promoter, and after this enhancer interact with the promoter to activate this gene. And for WGS, normal WGS data, we don't have this information, like the information of interaction. We have only the linear information, who is next to each other. But how HiC help us for this? If we assume this is as a part of the genome, as this is folded part of the genome, so normally here the blue line and the red line represent like linear region or next to each other region, but the HiC data doesn't include the only the red part or the blue part, but it is give us some paired in read which represent the interaction between this region, which is controlled by some protein. So here, out of this data, we will find that HiC library give us kind of information about the interaction between non-adjacent or distal genomic loci. So if this region, the blue one, we will have interaction between the blue here and the red one, which give us some reads about represent the interaction, which may represent some interaction way between enhancer and the promoter. Similar for read B and similar for read C. And based on this, usually we map this, we divide the genome into bins of some resolution, like 100 kb, for example, and we count how many interactions between these different genomic loci. And as a kind of representation, HiC data usually represented as a heat map or matrix. So if this, we consider this as the bend of the genome, this like the color scale represent the strong of interaction between different regions. And as we all expected that region next to each other by chance is expected to interact more with each other. But one of the main findings of the HiC studies that they divide or argue that the genome is divided into topological associating domain. So this topological associating domain represent higher order chromatin folding, and the main feature of this cells is we can say that genomic loci inside a TAD are interacting with each other more than interacting with region from the other TAD. So for example, if we take a look here, for example, this region, this you find that it is interacting with this region higher than interacting with this region, even if in the linear scale they are at the same difference, or sorry, same distance. So how this This was the main finding of high-C studies, and this was the first dimension of information we have. And the high-C protocol, as any NGS-based or sampling-based technique, are usually normalized for sequence bias like GC content and mammability. And the high-C correction method assumes that most of the genome loci should be observed the same or have the same frequency to be presented. However, this, based on our understanding of cancer cell line, this can't be fully corrected or correct because the widespread presence of structure variation, as we show about copy number variation. So for example, here, if we take the summation, row summation, we'll find that the average difference may be different. This for sure, this our like analysis or our way of explanation. But for check this further, we check the data itself. And even here, for example, if we took two cell line, one GM12878 normal cell line, and the other one is MCF7 cancer cell line. And this is the heat map representing the chromatin folding or the higher order interaction. In normal cell like GM12878, you will find that most of the region forming some pairs, but or some interaction with nearby region. But if we look here for MCF7 cancer cell line, you will find something weird a little bit, because if we look to this region, you will find that this region are interacting with all the genome. Here, for example, this same. And here also this region, this red or high intensity red, represents that this region are interacting more with other region, even it is not nearby. And plus to some increase of the intensity in this big region. The main question here is this data is correct, or this data we miss some information to accurately interpret this. To understand this, we check how the CNV is correlated with high-C contact map. And as we expect, we find that regions which have higher interaction or off-diagonal interaction, not to next to each other, are highly correlated with copy number event, like increase of some region or decrease in some region. As we can see here by the line here, of diagonal line, this represents that this region are interacting with other region, but mainly this is not the case, that there is some amplification or copy number gain here, similar to this region. And when also we did some statistics like this Spearman correlation, we find that the interaction frequency is highly correlated with the copy number compared to the other known systematic bias. So based on this, we make our computational solution for solving two main problems. One of copy number profiling, which consider as detecting both way of structure variation, large scale copy number variation, and focal alteration. And after this, correcting map, contact map correction. And for this, we developed two solutions: Sinatra, which is mainly for detecting the large scale and the focal copy number alteration, and high Sinatra, it is integrate the part of Sinatra for identifying the copy number, and after this, correcting the contact map. And as we discussed, we need to make this happen at the minimum condition, minimum information we mean, like from single sample data, we don't need to use matched or control sample. No barrier knowledge, because as we discussed, the difference between clonal variability and the alteration percentage change as we show for different cancer cell lines. So first, for this, Sinatra utilizes the RT signal for the WGS data for discovering first the large scale as we show here, and the focal alterations, estimating correctly the copy number, which is 2, which is 3, how many copy we have from each region. And this can also work in low coverage. And for this, we have little bit long framework. which to solve this hierarchical problem, first we utilize the main idea of us, or the main advantage of Sinatra is we use multimodal distributions to detect or identify the copy number, like what is the value of RD signal that represent the normal region. After this, we use some signal processing method like sophisticated filter and over segmentation for identifying all the change of the genome. Based on the copy number and this one, could identify the largest scale copy number variation, which we named as the isocopy numeric block. And finally, at each IB or each big region, we consider this as a population, and we model this using normal distribution to identify the focal alteration inside this, which we believe it is more related to gene expression. So the first part was to estimating the copy number reference. This is signal processing. For people or for computational guys, which usually work for signal processing or have information about communication system. This considered like a way of convolution. So we have the RD frequency distribution of the data, and we need to find which region here consider copy number 2, 3, 4. And what we did, we make shifting of this data and try to identify the region, that value which make the highest correlation or the higher matching. After identifying the copy number of 2, like for example, if we can, this because we know this data, so if this is the 2, for A427 here 3 and 4, the main benefit for this, now we have the interval. We can define the interval. So for example, the first interval here, I equal 1, this means copy number equal 1. It has interval from copy number equal 0.5 to copy number equal 2.5. And next, same like this, this is the copy number of 2, similar copy number of 3, and so on. And this helps us much because now we can do kind of over segmentation. So for example, if we consider this the segmentation way, we do segmentation of the data, we can know this all region are represent one region. This is not different even. They have some variation because of the coverage, the noise of the data, but mainly they represent one region. So here, for example, we consider as first IB, here second IB, third IB, and fourth IB, based on the region, big region at the same copy number interval. And after we define each IB, as we said before, that we use this region and model this using normal here with Gaussian model distribution to identify using a t-test to identify the outliers, like here this event, which this can be called as a focal amplification, and this can be called a focal duration. And Sinatra shows successful distinguish between large scale and the focal alteration. So for example, even we have many variation here, like some deletion, some amplification, small amplification here. It can identify this big as 1B deletion of CHB212. And similar for different cell lines, it can identify mycin amplification exactly based on the RD signal. And for benchmarking, we benchmark with 5 of the most used tools: TNVinator, Redepth, Free Climate, and MSIN. And we find that Sinatra is superior for identifying both LCV and focal alteration based on the precision, recall, and the F measure. So based on this, we now solve the first problem. From the WGS data, we could extract the information or the copy number information. So what next is how to use this information, which is copy number, to correct the high C data, so we can accurately interpret or understand the 3D genome conformation. And for this, what we did is the high Sinatra. First, we need to like to find the RD signal from the high C read, which is little bit different from WGS, and identify the copy number profile based on Sinatra approach, and finally correct the contact map from CN copy number bias as well as systematic bias. So for high Sinatra, our framework was in this, we can be explained in this way. What we all have, we don't depend on any other data. We have only high series. And for high series, we need first to compute the RT signal. And after this, we utilize the copy number equaler module from Sinatra to identify the copy number track and use this track to normalize the high C data. So our contribution in this high Sinatra tool is in these two parts: read depth calculation and contact map correction. So quickly again, if we go back to the comparing the high C data to the WGS data, high C data is different from WGS data because while genome sequence data, we have paired N read, which is distributed in kind of uniform way in absence of copy number in the genome. So we expect to have the same number of read or in different number of genome. But here, based on the experiment, we have only the reads next to some place called the restriction site, which we use. It is used in the experiment. So if we look to the high C data, we find that most of the reads are next to the restriction fragment or restriction site. And some big region here may don't have any reads. And for the other tool, they usually use large bin size, so it can use this. Like if I use large bin size, so I will count this for in the big bin size. But for us, we think in different way. We think this read, if I have some reads in this region or some reads in this region, this meaning they represent all of restriction fragment region. So based on this, we use this number of reads next to the restriction site for as a number of reads for the whole area, like the complete area here, or complete region like restriction fragment 1 and the restriction fragment 2, which show much better result. And for this, we propose this technique as called restriction fragment count approach for computing the RD signal. So for example, here, if we compare our restriction fragment count approach, counting approach with the other most used methods, one is called exact cut mapping and the other one called midpoint mapping, we will find that here the distinguish between the copy number event is much clear using the same number of reads. Here you will find that there are many regions or many bins which have zero count, as we show because of this problem region. And for midpoint, the dispersion is higher compared to our approach. So in this case, as we see, we can better identify the copy number event and is both large scale copy number event and focal alteration. And here also, if we compare by the RD frequency distribution or the copy number frequency distribution, you will find that here we have two peaks in the RD signal, which represent this big region and this region, which here it is overlapped more because of the distortion. And this helps us to identify the copy number profile from high C data as same way as the copy number profile from WGS data, and they give us the advantage of accurately profile the copy number and use it. The next part is contact map correction. contact map correction, we use Poisson-based generalized linear model. So we have here for high C data, if we talk about high C data, we have well-known three sequence or systematic bias called GC score and the mappability and effective length. This is usually used for normalizing the contact map or the interaction frequency. But what we did, it is can be considered as a simple but is effective. In a way, we say that copy number variation can be considered as one additional bias in the interaction frequencies or the heat map. So we add this copy number feature as a one of the bias to correct the interaction frequency based on the all systematic bias plus the copy number variation, and to normalize to remove this effect. And when we compare with the well-known or most used tool, there are three most used tools which are applicable for cancer cell line. One called iterative correction or ICE. This is the widely used high-C correction method, and the other one CAIC and 1D. These two are normalizing based on the approach that most of the regions should have same number of reads. And if we compare here, we'll find that from the visual comparison, we can say that High Sinatra and the ICE and the CIC, all of three have, in a way, good way of that most of the heads are in similar size, and they are on the diagonal, and these are some clear area of interaction, which they mean that both the three show similar way of normalizing, and 1D, which is similar to our approach, is that it is explicit approach that it is normalized based on the known type of the bias. It still have some of them. But the most interesting part that we find that if we after we normalize, we make the 1D signal variation, we take the summation of the all reads after the variation. So here, if this is the raw data, represents the summation of reads at each bin, or here the averaging, like 1D averaging, we'll find that High Sinatra shows the minimum variation. Here, shows the minimum variation between the different regions of the genome compared to the ICE and CIC, even if here they show from this visual representation, we are a little bit similar. And the most important part and interesting part, we find that implicit approach have one dangerous problem, that in some cases they do overcompensation, which this is usually part or usually exist in normalization. So if we look in these two regions, you will find there are flipping. Like in the raw signal, here is bottom, here is top, here is bottom. You will find it is inverted in the ICE and even in CIC, which this means that I do over normalization of overcompensation, because I don't know, or I don't need to know about the source of bias. I normalize to have all same number of reads. And this confirmed previous studies that show that implicit approach like ICE and CIC is not may lead to distortion in the high C interaction frequency, and show that our approach, High Sinatra, is the most suitable tool for correcting the high C data. And so for this, we developed Sinatra and High Sinatra for mainly detecting the copy number information from the same data, like WGS or high C, and after this, using this data to normalize the high Sinatra or high C data. So now, after normalizing, many clustering techniques are used, for detecting or identifying the topological associating domain, what's the interaction between the regions that highly interact with each other, or maybe also identify the interaction between some enhancer and the promoter. And this was the main idea during my PhD. And we here in my PhD, we connect that we try to accurate modeling of high C data using integrating of copy number variation to better modeling for cancer cell line. And after my PhD, I now focus in single cell analysis or multi-omics data integration because this is the emergent technology, and this helps in same approach like cancer accurate modeling data integration. So for single cell analysis versus bulk analysis, the main advantage is that in bulk we have the averaging of all the cells. So if we have many cells here, we will do like, for example, bulk RNA-seq, we will have the average gene expression from all the cells, and we may try to find to like differentiate between them or cluster them, but it is usually difficult to find subpopulation. But the single cell analysis or single cell approach, they help a lot in this way because they give us information for each single cell alone. So we will have now some cells or the gene expression of some of each cell, and we can do some machine learning or data science approach or clustering way to identify the subpopulation. And this helps a lot in identifying and characterize heterogeneous cell populations. Next, it can help also to discover new cell markers or regularity pathway and to find novel cell type or cell status and profile tissue under different conditions. And for example, one of the work we are doing now is to try to find how the macrophages are changed between cancer and normal cell lines, because we now can identify the subpopulation of macrophage. The interesting part, and in 2013, single cell data is represented as the methods of the year by Nature Method. So Nature Method at 2013, they say this is the method of the year. And as a result of this, this is the computational pipeline for the single cell, for example, RNA-seq, given that we have the cells and the genes, we have here the count matrix, which represents the interaction between or represent like how many genes are represented in each cell. And the pipeline for single cell is huge pipeline because now we are talking about multi-dimensional data. We have now, as we see, one matrix, two matrix, three matrix. This all for each cell we have like one array or one row vector. So it starts by quality control and normalization and doing some data correction for batch effect. And from here, we do like some visualization. So we use some dimensionality reduction, BCA, TSNE, and UMAP. And after doing like dimensionality reduction, we apply clustering technique to identify the subpopulations. So, and this is the part which need more biological knowledge because this is defined using the marker. So, which gene is well known to be represented is in subpopulation like macrophages, monocytes, and so on. And after this we do like trajectory inference, differential expression, and do this analysis under different conditions. So this big pipeline, which I feel or I believe also that it is the area which most of the computer scientists should now contribute at. And this results in hundreds of computational tools till now, like for different kind of problems, like from visualization, clustering, dimensionality reduction, normalization, and the integration here is 10%. And the integration, we can think about integration. They are integration between data from different different condition, or the most advantage one in integrating different type of data. So at 2018, science represents single cell integration as the breakthrough of the year. They were talking about integrating transcriptome or single cell RNA-seq from different experiments. And in 2019, Nature Method now talk about single cell multimodal omics. Before in 2013, they talk about single cell RNA-seq, for example, it is method of the year. Now we talk about integrating different multimodal data, like, for example, transcriptomic spatial analysis, uh, copy number, high-C data, attack-seq, shape-seq. So now we are thinking about more dimension of the information. So if we first say that, again, if we go back here and we talk that, we say that the interaction or the single cell data is like multi-dimensional, we think more about if we integrate this data with different data. Now is the time of integrating multimodal data based on, not based on my opinion only, but as we see here in Science 2018 and the Nature Method 2019, they show that integrating this data will give us more insight during for single cell for information. And as a conclusion, I believe, like personally believe, that normalization and integration are important for most of the biological data. In my work, we show that cancer cell line are associated with different biological phenomena, and we develop high-C natural to identify the copy number and to do kind of integration of this copy number of information to normalize the high-C contact map and follow our approach about how to data integration for accurate model of cancer. And I think also it is the time of integrating multimodal or multi-omics single cell data. And I think like next year, if I will be present in this event, I will show some computational solution or my computational solution for integrating single cell data or multi-omic single cell data, especially transcriptomic and spatial transcriptomic, or transcriptomic and attack-seq data. Thank you all for your attendance, and I am happy to share any question with you.
Thank you, sir. Sujay, sir.
Yeah.
Thank you.
Yes,
Thank you, Dr. Walil,
Yeah.
for your an interesting and informative presentation to us.
Thank you.
And actually, we have presented high Sinatra framework that has been developed to identify high resolution CNV profiles of cancer signs from IGC data and to explicit correct chromosome contact maps from systematic biases, including CNV association bias.
Yeah.
So
Yeah.
it is a very nice presentation, and
Thank you.
any participant has any questions you can ask. And after the session,
Pradita, one question is there from Orijit Bose. The question is, how have you reducing the biasing?
So here it is reducing the biasing for the normalization. We can say this is a generalized linear model. So here what we do is we first estimate the bias source, like by random, we estimate the relation between the counts. Like we have here, for example, we can say we have some counts here for each event, and for each genomic loci, we have the like GC content information, mapability, and copy number information. So we use them first to model the relation. So here, for example, if we have the interaction frequency and we have the information of bias like GC, mapability, effective length, and what is called the copy number. So we use them first using generalized linear model to estimate these values, which is the parameter of the model. And after this, using this value, you can estimate the value based on the bias only and divide the interaction frequency, the row of interaction frequency or the observed interaction frequency by the expected one. So mainly we try to model the expected interaction frequency for each genomic loci and divide the expected one, the observed one by the expected one to find the normalized copy number variation. The normalized interaction frequency.
Sir, Professor Joya Bandyopadhyay from our university has raised your hand.
Joy Bandhavadhyay, already raise your hand. So please. Joy Bandhavadhyay, please.
Ma'am,
Madam,
please ask.
yes.
Yes.
Yeah.
Yes. Good evening. And I am Jaya Bandyopadhyay from the Department of Biotechnology. And Dr. Khalil, it was really a very excellent lecture. And my question will definitely not be directly to any particular problem in or any question from the work, but I have an overview and a question which would be actually addressing your background. And I'm very amazed that you are from an engineering background,
Yeah.
as I understand from the introduction that our professors have made earlier. And then you have nicely portrayed all the biological data, wherein actually you have brought in the cancer subject and compared the data, means what is a cancer line or what is a cancer cell, how the DNA, how the genome is different from the normal cells. You have made pretty a brief, but yet you have kept it quite concise and made it very clear to our students. Especially, I am more concerned with the audience, which is having the students in large numbers. So, Dr.
Yeah.
Khalil, in your journey from an engineering background to this biological data analyst, how difficult was it for you to really cope up with the knowledge that you had to gain through your research, and then finally you have come up with such excellent computational work which you presented? Because this is, I am curious why, because there are students from both the backgrounds. Hopefully, there are some students from my department and from my field. And definitely, since this webinar has been arranged by the computer science department, so there would be expected there would be a lot of computer science students and information technology. So, my question is, how difficult was it for you to make use and make the best out of your background to channelize into this kind of a research? And if you can share a little bit of your experience, then it will be a kind of an interest to the students who would later take up these kinds of research in the near future. So this is my
Okay.
curiosity, and I'm really thankful that you had brought in so many aspects of the modern science and the highest technology that is now being implemented in the field of in the biological science. So, Dr. Khalil, I'm really curious, and thank you
Okay.
for allowing me to share my views with you.
Okay. Thank you a lot for your words. And my journey was, it is, we can say that my journey started 5 years ago when I first joined NTU. Before NTU, I was only work in computational, like computer science or computer architecture, and I have background for communication system, signal processing, and computer architecture in a way, or computer science. But when I joined, it was difficult for me first to understand the main biological concept, but the main interest with, or the main point that make me interested in the field, that in compared to our like communication on computer architecture, in computational biology we don't have the answer, we don't have ground truth, we try to find the solution for a problem that we don't know it is the correct solution or not. Like we are overthinking or keep thinking about how this data is correct or not, not compared to my field. And the main part which helped me a lot, to be honest, and like i I think, or I recommend any student or any young researcher to do, is to work in like in deep connection with biologists. So I have one supervisor in my PhD thesis is Prof. Marta Saniel from School of Biological Science from MTU. He is biologist, and he was the one who gave me the much knowledge I need. And so the discussion usually was between me and him. He has the biology background, and like teach me the biology background, and I have the computational solution. How to match this to this? I think the benefit or the most important part in computational biology, it is multidiscipline, like the introduction said today, that we have statistics knowledge, we need computational knowledge, computer science, data science, we have biology. All this, no one can have this at the maximum level to do all this together. But as a work, collaborative work with my with a professor, Marta Saniel from School of Biological Science, he gave us the knowledge I need to formulate or to can imagine the data, what the correct biology, and match this with the data based on my knowledge. So it, for me, to be honest, that in some, like in the second year and the third year, I thought it may be not be over, I may not finish my PhD, because it's difficult to get information from this old field. It was tough for me at the beginning, but after I gained the knowledge, I think now it is, I keep going, like as I told that in my future or next year I will join a lab to work in single cell data integration, because of the knowledge of I gained from the communication with Dr. Emarta. And one important note to say that I didn't take any biological course. This all is self-learning and by discussion with Prof. Emarta. I didn't have any biological course during my bachelor, during master, during PhD. Don't have biology course, but I study this like from books and online, and discuss mainly with Dr. Marta, which helped me a lot to connect the lines of biology there. Thank you.
Oh, thank you. And that means that if there is scope for the computer science department later on, what I understand from your talk today, that you have covered a large not many topics, and it actually would be better that if you could propose, or if the our university can propose some workshops which would definitely help the students later on if they have any plans in the near future. I think your expertise will definitely be a great help for the students who are pursuing bioinformatics in general, and also those who are using computational skills in their research in the field of life science. I think your type of work can be programmed with some workshops, and I think Dr. Mistry or Dr. Sharkar later on it is my suggestion, humble suggestion, that you might consider to have some workshops with Dr. Khalil in the future, which will surely benefit the students of our university. Thank you so much. Thank you.
Thank you. I will be happy
Thank you,
to
ma'am.
do this, and we may plan it for especially for single cell, because this is like
Absolutely.
as I told, this is
Absolutely.
the emergence technology. Any
Absolutely.
computational guy which would like to go to this field now, I feel this is the most important, and this not only my feeling. This is also can be confirmed by the science and the nature methods, like paper which say this is the current breakthrough in the field of computational biology.
Yes,
And
yes,
thank you.
absolutely.
Okay.
Absolutely.
Thank you,
Thank you.
Madam, for your
Yes.
good and
Yeah.
narration.
Yeah,
Okay.
yeah. Always welcome. Always welcome.
Okay, thank you, Dr. Khalil, for your nice and very informative presentation to us. Actually, this
Thank
is
you.
very important, and actually, we want in future your physical presence in our university to organize some webinars or some workshop or some
I will be
FD.
happy to do.
Yeah. So
Yeah,
actually,
for
we will
sure I will
invite
be happy to do this.
everyone in India, in our university, to join, and very nice. Actually, we need your presence in our physically, not digitally. Okay, we need your physically in our university
Yeah,
after pandemic situations.
yeah, I
So
like to be more physical interaction than as digital interaction, and I hope COVID-19 will be over soon, and for sure I
yeah,
will be happy to come to India for presenting this work.
yeah. So we will invite you. Okay, so we can obviously come to India
So,
and university to give your nice attitude and presentation to the students and our. Okay, so thank you, Dr. Khalil,
thank you.
agents in this webinar, and welcome to all of us. Okay.
Pradutta, shall I include something?
Yeah.
Uh, yeah. About the Joya Madam's suggestion, ma'am, actually, I and Pradutta already before the pandemic situation, one workshop already approved by the sir, by Vice Chancellor sir, and all the things we have already done that time actually. The funding and everything already that time already approved, and this webinar actually done that time in a 5-day workshop. But the pandemic situation, we just go through this half day webinar. So in
Oh,
our
I see.
perspective,
Uh,
yes, and then all the this will actually in past we have think of a workshop, and that workshop actually approved by the honorable Vice Chancellor sir. So that's why.
but it was not held, right? It was not held.
Yeah, ma'am. Yes.
Actually, if what I understand from today's from his talk, and also from the earlier talk by Dr. Barik, it is definitely it is organized by your department. But I think
Yes.
our students would be more interested, and I think if they are there, they must have gained a lot of knowledge, and that is why my question was that if a person from an engineering background can have so much of this data to analyze with the biological data, how can the students vice versa from the biological field to the engineering field, or the engineering to the biological field? It would have been more interesting. It is just a suggestion. It would have been more interesting because both of the speakers today had a lot of biological data. A little bit of simplified diagram in your talk would have really brought in some interest in even in the engineer's mind, because sometimes it could be a little bit difficult for someone to understand the jargons, and that is what had been bothering me all through this. And I pardon me if I'm wrong, but it had been bothering me. How will the students cope up with those jargons? Means you can understand, Dr. Khalil, what I mean by the
Yeah,
jargons,
for
the
sure.
biological jargons, and repeatedly they were there in your talk, but it is so much of data that there was no scope for you to explain in details, not possible in this short time. But
Yeah, for sure.
yes, having said that, a workshop will help all the students from all the various fields across the fields to nurture their minds into the different fields. And I'm looking forward to those kind of workshops wherein the expertise of such speakers like you today have been invited by the engineering department and deliver talks mostly related to the biological data. So thank you
Okay.
so much. But
Okay,
I think it was a good
okay,
attempt.
we are behind the schedule. Okay, so there
Oh,
are
sorry.
Thank you. Thank you.
I'm so sorry. I'm so
Thank you,
sorry.
sir.
Okay.
Thank you.
Thank you
so
so much. Thank you.
Thank
Okay.
you.
Dr. Khalil, okay, for your presence in this webinar. And
Thank you.
now the next speaker, eminent speaker, Dr. Shivnath Koyal. Dr. Shivnath Koyal is currently working as an associate professor in the Department of Metallurgical and Material Engineering, Jindal University, Raigad, India. Dr. Koyal completed his PhD in Material Science and Engineering from NTU Singapore in 2011. He worked as Senior Product Development Engineer in Advanced Micro Devices Private Limited, Singapore. He also worked as a postdoctoral research fellow in NTU Singapore from 2013 to 2019. Dr. Koyal research work focuses on synthesis and property evaluation and advanced material for biomedical applications. And various international peer journals presents. Am I audible?
Some problem?
Sir, your voice is breaking. There is some, I
Yes.
think, network issue.
Uh,
So, yes, sir.
am I audible now?
Yes, sir. Yes, sir.
No.
Please continue, sir.
Actually, Dr. Koyal is, is the recipient of prestigious silver medal awards for the best MTech student in order of merit from IIT Kharagpur and PhD fellowship from NTU Singapore. Dr. Koyal has published his research work in various international peer-reviewed journal and presentation in international conferences. The title of his talk, Magnetically Guided Smart Nanocare-Based Targeted Drug Delivery System. Well, Dr. Koyal, am I audible? Am I,
Sometimes the voice is
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
so, so Dr. Koyal, please present your presentation.
Yeah. Thank you, Dr. Pradyut Sarkar, for your nice introduction about me. Am I audible to everybody?
yes. Yes.
Yeah.
Yes. Yes, sir.
Yes, sir.
And can you visualize my
Yes, sir. Yes, sir.
slide?
Yes, sir. If you can make it in slideshow view.
Okay.
Yes, sir. Yes, sir. Please.
Okay, good evening, everybody. First of all, I would like to thank the organizer, Maulana Abdul Kalam Azad University of Technology, for inviting me and giving me the opportunity to deliver a talk in this webinar. It's really an honor for me. As Professor Pradyut mentioned that I'm currently working as an associate professor in the Department of Metallurgical and Materials Engineering. Today, I'm going to give a presentation on the topic magnetically guided smart nanocarrier-based targeted drug delivery system for cancer therapy. As my previous speakers, they just already enlightened about much more insight about the cancer cells. So now actually my topic will be much more focused on the therapy, cancer therapy. how we can use the smart materials to target the drug directly to the tumor site. So basically, I'll cover this following points. What are the present challenges in drug delivery system, and what is the importance of or utility of magnetic drug targeting, and this is the technological solutions where we can implement, and the objective of this work and what is the significance of work. Then in methodology, actually, what are the materials we have used for this topic, actually, that synthesis part and then characterization part. Then some biological part I'll just put for the in vitro test, like drug loading, especially doxorubicin drug loading and release study. And then I'll, as this is organized by computer department, so I'll also show some simulation, actually, that for that targeted drug delivery, and I'll show some results and discussion, and then finally I'll draw the conclusions. So as you know that cancer treatment is a key challenge in biomedicine, with more than 10 million new cases every year. Global cancer rates are expected to double by the year 2030. According to the report from the International Agency for Research on Cancer, a branch of the World Health Organization. The statistics for the global incidence and mortality rates for major types of cancers are shown in the figure. And from the figure, you can easily visualize that in developed countries, cancer is the second biggest causes of death after cardiovascular disease. The situation is also alarming in developing countries. Some cancers are more common in developed countries, as for example, prostate, breast, colon cancer. And cancer is characterized by the population of cells that grow and divide without respect to normal limits. And these cells invade and destroy adjacent tissues, and that may spread to distant anatomic sites through a process called the metastasis. And cancer is caused by a number of factors, such as unhealthy lifestyle, inadequate diet, exposure to carcinogenic. So these are the different causes. And there are different types of cancers according to their localizations, cell type, malignancy. But whatever the type, all that shares actually a common trait. If undetected and untreated at an early stage, the risk of death is very high. Now, what is the current method of cancer treatments? The most we know that surgery, radiotherapy, thermotherapy, and chemotherapy. However, the existing conventional cancer therapies suffer from numerous drawbacks. Let's say, example, surgery is an invasive method by which tumor is physically removed from the body. There is a risk of complications during the surgical process, and also the possibility of post-operative problems such as pain, infection, bleeding, blood clots, and slow recovery of organ function. Radiotherapy involves exposing cancer cells to high energy radiation such as X-ray, gamma rays to kill cancer cells. However, radiotherapy is non-specific in nature. That means it destroys healthy cells as well as the cancerous cells. Therefore, the side effects from radiotherapy include fatigue, damage to the skin, lungs, digestive tracts, mouth and throat, changes in brain function. And in chemotherapy, highly toxic drugs, cytotoxic drugs are injected intravenously and transported to the tumor site through blood flow. This treatment involves a schedule of regular administration of drugs distributed over several days. The major disadvantages associated with this chemotherapy is the general systemic biodistribution of drug throughout the body, which results in insufficient drug concentration at the tumor, therefore decreasing the effectiveness of the treatment. More importantly, these cytotoxic drugs are highly non-specific in nature. That means they kill both healthy cells as well as the tumor cell, resulting in serious side effects. And these side effects include hair loss, nausea, diarrhea, infertility, cognitive impairment, heart damage, nerve damage, liver damage, lung damage, etc. So therefore, it is very much important to selectively target the chemotherapeutic agents to the tumor site as precisely as possible in order to reduce the systemic distribution of cytotoxic drugs and to reduce the associated side effects. Now, the advantages of this targeted drug delivery are that the targeted drug delivery can improve the outcome of the chemotherapy by allowing the maximum fraction of the drugs to the tumor cancer cells without the adverse side effects to non-targeted sites. Now, in this aspect, magnetic nanoparticles, which I'll mostly cover in my subsequent slides, they are very much important and they offer very attractive possibilities in the field of biomedicine. The magnetic nanoparticles have their sizes smaller or comparable to the cell or a virus or a protein or a gene, and magnetic nanoparticles that can be functionalized by either some polymeric materials or some inorganic materials to which drugs can be attached, and that can be driven by an external magnetic field gradient. And that penetrability of this magnetic field into the human tissues enables such functionalized magnetic nanoparticles to deliver drugs, anti-cancer drugs, to the targeted region of the body, such as tumors. And also magnetic nanoparticles that can be also heated using some alternative magnetic field that results actually moderate degree of tissue warming, and that also causes some cell destruction, cancer cell destruction. And this method of actually heating is called the hyperthermia of the tumor. Magnetic nanoparticles, as you know, that also can be used as a contrast enhancement agent to improve the resolution of the tumor imaging in magnetic resonance imaging, you know, all MRI. So that means this magnetic nanoparticles, it has huge potential for the actually in terms of detection in form of MRI, in terms of the therapeutic, that means for the targeted drug delivery, and also magnetic hyperthermia, that means the heating of the cancer cells. So you can use this magnetic nanoparticle as a multimodal approach of cancer therapy. Now, in magnetically targeted drug delivery, what actually is done actually in magnetically targeted drug delivery system, a magnetic nanoparticle carriers, okay, that are targeted to the tumor site by an external magnetic field, okay, and this method can be a promising alternative to solve whatever the critical issues that are associated with the conventional cancer therapies. Now, in magnetically targeted drug delivery, magnetic carrier particles loaded with anti-cancer drugs that are injected into the patient body via the human circulatory system. And these magnetic carrier particles that are localized at the tumor site by applying an external magnetic field near the tumor site. And drug release occurs following the localization of the magnetic nanoparticles at the tumor site by changes in the physiological condition, such as pH, osmolality, or temperature, or some different enzymatic activity. And the released drug is taken up by the tumor cells. So therefore, this magnetically targeted drug delivery increases the drug concentration at the tumor site and minimizes the distribution of drug in the healthy tissue. So these are the advantages over the systemic chemotherapy. Okay. Now, the objective of this study was to determine whatever the conditions for the effective magnetic drug targeting. As I already mentioned before, that this is involved that your drug loaded magnetic carrier particles you have to inject. So that means you need to use the some ferrofluid system, human circulatory system. So that means there are different parameters actually, such as blood flow rate. And as you are concentrating the magnetic nanoparticles to the tumor site by applying some external magnetic field, so that means that magnetic field strength and gradient, that also plays important role. Next is the magnetic properties of the carriers and the distance of the tumor from the magnetic field. So there are different parameters actually, and I'll subsequently discuss. So whatever the parameters you can play to find out the whatever the optimum conditions, so that you can make the efficient targeting to of the chemotherapeutic drug to the tumor site. Okay, the significance of this work is the integration of the synthesis, characterization, and property evaluation of magnetic carriers, and establish the physical parameters and material properties for effective magnetic drug targeting to tumor site. As I'm from the material background, so I just want to integrate all these aspects for some practical solution of the biological aspect actually. Now, the material systems, we have chosen magnetic core material because we have to, I already mentioned that we have to choose the external magnetic field. That means you have to choose the materials which have some magnetic property. So we have used iron oxide and iron nanoparticles. This actually magnetic particles we have used as a core material because it's already established that these magnetic particles are suitable for magnetic drug targeting, and they are non-toxic in clinically relevant quantities, and they are quite routinely used as MRI contrast enhancement agents. And then coating materials, because uncoated particles that we cannot use because that cannot absorb the drug. So we have to use some coating materials, and that coating materials we have chosen polyvinyl alcohol and also gold. And polyvinyl alcohols, it's a very good biocompatible, and it is also hydrophilic in nature. They are also biodegradable, and this polymeric polyvinyl alcohol, it can be easily functionalized. And gold also is very biocompatible, and it is also inert in nature. And gold is very good surface chemistry, and it has also biological reactivity. And anti-cancer drug doxorubicin we have used for the drug targeting, drug release, and drug loading study. And this role of coating that actually stabilizes the nanoparticle in biological suspension, and that provides the interface for functionalization with the drug, and that also avoids immediate uptake by body's reticuloendothelial system. So now that's the materials actually we have synthesized using that chemical synthesis route. So basically, we have synthesized that iron oxide nanoparticles by choosing the ferrous chloride and ferric chloride. This actually you have to choose in a 1:1 molar ratio, and that was mixed in aqueous solution. And then with that aqueous solution mixture, sodium hydroxide solution was added drop by drop at 80 degrees centigrade with constant stirring, and black precipitate of iron oxide particles was formed. Then another particles, actually gold coated iron oxide nanoparticles in situ, actually we have synthesized by using reverse micelle technique. So basically it's two different micelle solutions was chosen. One is your ferrous sulfate in the micellar solution is in using cetyl trimethyl ammonium bromide and butanol and octane, and sodium borohydroxide, another micelle solution. And this sodium borohydroxide actually acts as a reducing agent, and that mixed together. And another micelle solution actually we have used actually gold solution and tetrachloric acid, and we have used sodium borohydride as again as a reducing agent. So basically the idea behind this choosing this technique that sodium borohydride actually first reduces ferrous sulfate to iron, and whatever the sodium borohydride, that also reduces the gold actually +3 state to 0 state, and in situ they actually form the coating of the gold on the surface of the iron. So in this way, we have formed the gold coated iron nanoparticles. Now, in experimental procedures, we have used different techniques to assess that what are the actually synthesized materials we have formed. So basically, the structural analysis and size of the morphology analysis of the particles that is done by transmission electron microscopy. As I already mentioned, this is the nanoscale particles. So we have to use some high resolution device to identify that whether this is what is the size. And XRD we have used for that is actually structure analysis. Then we have used some polyvinyl alcohol, and in order to determine how much amount of the PVA that has been coated, so we have used thermogravimetric analysis. As our materials is magnetic, so we have to study the magnetic property, and this magnetic properties was studied by VSM, vibratory sample magnetometer. And then drug loading and release study, actually that was performed using UV visible spectroscopy. And the drug binding, that means this doxorubicin drug, how it actually interacts with our actually material system. So that we have also already studied using FTIR analysis, Fourier transform infrared spectroscopy analysis. And then in vitro test we have done using magnetic carrier particles targeting, as well as drug loading and drug release study. And then actually we have also done some modeling study using CFD modeling for ferrofluid transport phenomena in deposition of the blood vessel. And we have actually analyzed different parameters involved for this magnetically driven targeted drug delivery, including magnetic carrier particle size, what would be the effect of the blood flow rate, then magnetic field strength and gradient, and magnetic geometry. So now I'll show some of my results. First, gold-coated iron nanoparticles. As you see, the morphology of the particles by transmission electron microscopy. So basically, uncoated particles actually they are homogeneous size distributions, and this particles average size of uncoated particles, that means iron, is 10 nanometer size. For gold-coated iron nanoparticles, the average size is around 15 nanometer. Next, the XRD analysis to identify what are the actually synthesized materials phase. And as you can clearly visualize, for uncoated samples, actually, that means there is no gold coating. All actually the particles that are oxidized to iron oxide. That means no parent actually, that means no iron as such is remaining, so it is actually oxidized. But when we have used the gold coating, you can easily see from the XRD analysis that gold and iron peaks that are actually present, and their corresponding actual reflection planes I have already indexed, and they are subsequently the selectron area electron diffraction patterns. And the right hand side figure, you can see how this gold and iron actually reflection planes look like. So from this actually analysis, we can conclude that without gold coating, actually that iron nanoparticles that are oxidized to their corresponding oxides. So basically Fe3O4, they are converted to Fe3O4. Now the magnetic property, you can see actually, so this is, I already mentioned that these particles are all magnetic in nature. So we have to study the magnetic property. And you can see all actually the nature of this curve is that super paramagnetic nature. That means they have zero coercivity. That means the interesting things is very important for the magnetically targeted drug delivery. That means whenever you apply the magnetic field, that time it behaves as a magnetic. And when you remove the magnetic field, this acts as a diamagnetic. That means there is no means remnants that are present in the magnetic field. So that's why it is called the super paramagnetic nature. And that is very useful for our application in the targeted drug delivery. And the most important thing is that that you can see with coating of the gold, the magnetic property decreases. You can clearly visualize from the curve. Next, we have analyzed that how actually this synthesized materials behave with the doxorubicin drug. And as I have already mentioned that we have used the doxorubicin as a anti-cancer drug, as a model drug. And you can easily see from the left-hand side that from the FTIR analysis, you can see the doxorubicin drug shows a band at 3450 and 3330. The 3450 band is basically from the amine actually primary amine that actually present in the doxorubicin structure, and 3330 is from the hydroxyl stretching vibrations. Okay, and whenever you we have actually conjugated this doxorubicin with gold coated iron nanoparticles, you can see from the FTR analysis the peaks are actually at that region that are overlapped, they are broadened, and they are actually switched to some peak shape actually at 3340. So that means this peak broadening actually that confirms that actually the doxorubicin attached to the gold coated iron nanoparticles by the interaction with the amine group of the doxorubicin with the gold. And this gold also you can see how again UV visible spectroscopy, you can pure doxorubicin drug that actually gives you plasmon resonance and some particular wavelength, that is 485 nanometer, and also gold coated iron nanoparticle, it actually shows that your the peak at 520 nanometer. However, there is a rate shifting of the surface plasmon resonance, okay, when you add actually doxorubicin to the gold coated nanoparticles. So with these two analysis, we conclude that this actually the doxorubicin is attached to the gold coated iron nanoparticles by interaction with amine group of the doxorubicin with the gold surface. Now this is the actually drug loading and release profile. You can see on the top figure the doxorubicin drug loading behavior. You can clearly visualize the with increasing concentration of the gold coating on the there is a drug loading capacity increases, and the bottom figure shows the drug release profile, and that shows that maximum 18% and 25% of the adsorbed drug that are released from the carriers. Okay, so this is the drug loading and drug release profile of the gold coated iron nanoparticle. Next, we have actually studied this in vitro targeting. So basically, how much actually these magnetic nanoparticles that are retained using some different parameters. That means with the change of fluid flow rate, different fluid flow rate, and also we can have changed the distance of the magnet from the distance of the actually the targeted regions. So we have changed at various distance, and by changing this various distance, we have also changed the magnetic field strength. And you can clearly easily visualize from these two plots that the magnetic retention, that means the magnetic capture efficiency of the magnetic carrier particles, that increases with decreasing fluid flow rate of the actually fluid, with increasing magnetic field gradient, the retention increases, and with the increasing saturation magnetization of the magnetic particles, the capture efficiency increases. Next, I'll show the results of the PVA coated iron oxide nanoparticles. You can see from the TM micrograph that uncoated iron oxide particles, the particle size is around 12 nanometer. For the PVA coated iron oxide nanoparticles is around 14 nanometer. And this is again the XRD analysis to assess that what is the exact phase that it is formed. You can see that from the left, actually the bottom diagram, that all the peaks actually uncoated is the Fe3 over particles. But the interesting fix, all the peaks actually that becomes at the same positions. But with increasing PVA concentration, there is a peak broadening. Actually, gives you to the reduction of the crystallite size. And also we have measured whatever the corresponding crystallite size by actually refinement using some red field refinement, and we have found that the crystallite size is around 8.4 angstrom. Now the left-hand figure, top left-hand figure, that actually gives you idea from the TGA analysis that how much PVA that has actually that has been coated to the uncoated Fe3O4. And how we have analyzed, actually basically you can see uncoated Fe3O4, there is flat, almost flat in that temperature range from 200 to 400 degrees centigrade. But for the PVA coated iron oxide nanoparticles, you can clearly see there is distinct weight loss from 200 to 400, 200 to 400 regions, and that is basically due to the decomposition and subsequent collapse of the bonding of the polyvinyl alcohol. And from this actually zone, we have measured what is the corresponding weight loss, and this weight loss actually gives you the amount of polyvinyl alcohol that has been coated with the on the Fe3O4. And then we have also tried to identify how this PVA actually is attached and means the binding have bound to your uncoated Fe3O4. And then we have from the right hand side figure, the your FTIR analysis, you can see that there actually the bands at 2912, 1416 and 1019. Okay, that actually confirms the attachment of the PVA to the iron oxide nanoparticle. Same as this is the magnetic material, so we have performed the magnetic measurement using VSM. So basically, you can see again with increasing PVA concentration, the magnetic property actually decreases. And all magnetic particles, they are super paramagnetic in nature. Okay, and these supermagnetic particles are very useful for our desired magnetic targeted drug delivery. And this actually figure shows that doxorubicin drug loading and drug release profile. So you can see that around 58 actually microgram loaded per milligram of carrier particles, which contains the 5% PVA. And out of that, maximum to 45% drug were actually released from the coated of 5.5% of PVA, PVA coating actually. And this also FTI figure actually that shows actually how actually this doxorubicin drugs actually is bound to actually this carrier particle. Carrier means PVA coated iron oxide nanoparticles. And again, we have found the same conclusion that it actually the doxorubicin drugs actually interacts or conjugates with the PVA coated iron oxide nanoparticles via the interaction with amine and OH groups of the doxorubicin with OH group of the polyvinyl alcohol. You can clearly visualize this from this pics here. This is also that in vitro targeting of PVA coated iron oxide nanoparticles, and how much actually carrier particles that are actually captured with the help of external magnetic field with the as a function of magnetic as a function of the fluid flow rate using different magnetic field strength and field gradient. And you can clearly visualize from the curve that this capture efficiency of the magnetic carrier particles that increases with decreasing flow rate of the fluid, with increasing field gradient of the magnetic, and with increasing saturation magnetization of the magnetic carrier particle. And this actually show the in vivo magnetic drug delivery, but this was actually done with Singapore General Hospital and with the help of the oncologist. So actually we have actually whatever the material samples actually we have actually given to them, and they have performed So basically, they used a buffalo rat and they actually cultivated some hepatoma 777 tumor incubated, and whatever the materials we have actually synthesized, so they actually make some ferrofluid and they inject it into the magnetic injected into that into the intravenous with the rat, and they have positioned some external magnetic field outside of the liver. And you can see in the right hand figure, the before any magnetic actually when there is no pre-injections, there is no actually of in the MRI study, there is no actually particles that remain in the liver. But after actually 30 minutes of injections, you can see clearly they have performed the MRI study, and they found that there are some occurrence of the magnetic particles. They are present in the liver of that liver region, that the tumor region in the liver, and they actually so that means so that this magnetic targeting that actually that can actually concentrate or that can target to the to your targeted region by using of the external magnetic field. Now, as I already mentioned that this the magnetically targeted drug delivery, but however there are different parameters that are involved. So it is not so straightforward as I mentioned you that you just focus some magnetic external magnetic field and then you can inject and then your particles will be just in that tumor region. There are various parameters that are involved, and we have tried to find out what are that actually parameters. Now, coming to the modeling part, so that's why we have actually to figure out what are the parameters and what is the effect of that parameters for this efficacy of this magnetic targeted drug delivery. So that for that actually we have used some permanent magnet. You can easily visualize from that model. So we have used some, let's say, as a vasculature of the tumor, and you just put the magnet at a distance or some particular distance, and there is some fluid flow. You can clearly visualize this is the inlet channel where the fluid actually flows in that x direction. And we have used different models, actually convection diffusion models, and we have used this our magnetic particles. So that means there will be some particle velocity as well, and we have used the Navier-Stokes model equations to identify what it would be actually the effect of the fluid flow, and with the help of the magnetic field strength to the near the vicinity of the magnet. That means our magnet should be positioned near the vicinity of the tumor site. Okay. So you can see this is the magnetic field strength and magnetic field gradient profile according to the x direction and according to y direction. But it is important to know that, as I already told, that our magnet field is positioned so that y component of the magnet is actually very important, because that actually gives you the driving force to capture the magnetic particles towards itself, and where the x direction, that is actually governed by the fluid flow. So this y component you can see. So we have actually performed two different magnetic geometry. One is your rectangular magnet, and one is the cylindrical magnet. And this you can see that basically the gradient, y component gradient. So basically this the gradient is maximum at the corner for the rectangular magnet, whereas for the cylindrical magnet, the gradient is maximum at the center of the magnet. Okay. Now we have performed some numerical simulation to give you the idea how this actually the flow rate, how is the magnetic field gradient, and how is the fluid flow rate that impact about the deposition of the magnetic carrier particles at the tumor site. Okay, so you can clearly visualize that when it's very time is short, this starts to build, okay, near the vicinity. Now, when the time is high, so that time you can see there is a clear profile of the deposition. And one interesting feature is that when you just increase the flow rate, okay, so when the flow rate is low, so there is clearly visualize, you can clearly visualize they actually uniformly they are deposited. But when the flow rate is maximum, when we increase from the 1 mm second to 40 mm second, you can see it is actually spreading. That means the deposition, they are actually the convective force of the fluid, they actually dominate over the your magnetic actually flow, magnetic the force. And that time actually the magnetic particles, they are sprayed to the at the region. Okay, so that's why we have to optimize the time as well as the magnetic the fluid flow rate. Next, we have used at different distance actually. So in order to figure out what is the effect of the magnetic field strength, as you know, when we increase the distance from the targeted zone, that means if the magnetic strength actually is decreasing. As you go nearer to the your target region, that means your tumor site, so that means the magnetic field strength that increases. So you can clearly visualize from the left hand figure, there is a higher deposition in case of stronger magnetic field. But when you just on the right hand side figure, when you increase the distance, that time your magnetic field strength actually decreases, and that time actually the deposition actually dispersed. Okay, so that means the convective effect actually, so there is actually a competition between these two forces. One is your magnetic force and one other is the convective force, and that actually ultimately gives you actually the what is the efficiency or what is the capacity of the deposition near the tumor site. Now we have also identified at the very high when the magnetic field strength is very low. Okay, so when the magnetic field strength is very low, and when is a sufficient actually velocity also fluid, you can see there is the vortices are formed. That means they are not actually deposited actually at the wall of the your vasculature. So that means what happens here. So actually this convective effect is very much predominant at this juncture, and therefore dislodging of the ferrofluid from the bottom wall to the center of the vessel. And there actually in this case actually you can see there is actually spreading actually. So there is no significant deposition at the particular target zone. So they actually flow. Now another interesting features we have observed from this simulation study, the effect of the magnetic geometry. As you can see from the left hand side, we have for the rectangular magnet, the deposition is actually at the corner of the magnet. Okay, because as I already mentioned that at the corner side, the magnetic field is predominant for the rectangular magnet, whereas the magnetic field strength and magnetic field gradient are maximum at the center. And you can clearly visualize in our simulation also, the deposition is actually at the center for the cylindrical magnet. Okay, so in that way, so by using the magnet, so where is the profile, how the deposition profile you can clearly identify. Okay, so now I come to a conclusion about my presentations. So basically gold coated iron nanoparticles and PVA coated iron oxide nanoparticles, they have excellent potential for tumor targeted drug delivery because they have a very high capture efficiency and also quite significant of drug that can be released. But the efficacy of this capture efficiency that depends on the site and the magnetic properties of the magnetic carrier nanoparticles, then the blood flow rate and the magnetic field gradient. The convective effect actually dominates over the magnetic effect in weak magnetic field, and magnetic deposition that actually near the corner of the rectangular magnet and the center of the cylindrical magnet. So these are the parameters we can optimize. So in order to find the efficacy, how we can effectively concentrate the drug to the vicinity of the tumor. That's all about my presentation. Thank you all for your attention.
Actually, we are beyond the time schedule. If you have any questions, you can ask me later after the webinar. Okay. So thank you, Dr. Shivnath, for your nice presentation and innovative, informative idea which you have given to us. You have delivered a lecture on magnetic drug targeting.
Yeah, thank you, Professor.
Magnetic carrier particles is an efficient method to localize drugs at the tumor site. So this is very, very interesting and efficient lectures for us. And in future, we need your physical presence in our university. Okay, and we'll invite you, and you can accept our invitation, and we'll come to our university for deliver your valuable lectures. Okay.
Thank you, Professor. Definitely should. In future, if there is a opportunity, definitely we'll collaborate.
Okay. Dr. Koyal, for your nice presentations. And now the last eminent speaker, Dr. Shaugato Hajra. So please welcome Dr. Shaugato Hajra to your presentations. Dr. Hajra now is an adjunct and assistant professor in IIT Roorkee. Actually, he has a lot of. Actually, I want to. Am I audible?
Sometimes
Voice is breaking.
voice is just,
Yes.
yeah.
So please continue, sir.
Yeah, am I audible now?
Yes, sir. Yes.
But video is not properly. And Dr. Hajra, he has completed his BSc from Calcutta University and MSc from Biophysics and Molecular Biology from University of Calcutta. After that, he completed his MTech from IIT Kharagpur and the topper and gold medalist in MTech in IIT Kharagpur in 2004. After that, am I audible?
Yes.
Yeah.
Yes, sir. Yes.
Please continue.
He has doing his PhD from University of Illinois at Chicago, USA, 2010. After that, he has joined as postdoctoral research fellow at St. George Children's Research Hospital, Memphis, USA. And then he has joined as postdoctoral research associate in Albert Einstein College of Medicine, New York, USA. He has hundreds of research papers on journal and conferences. He had 12 crore of research fund projects in your hand till now, and he has 3 patents. His research area on computational biology, MD simulation, virtual scanning, molecular infections, structural biology. So this is very important, and Dr. Shaugato Hajra is an efficient, eminent speakers. So welcome, Dr. Hajra. Please present your presentation now. So welcome, Dr. Hajra.
Am I audible?
Yes.
Yes, sir.
Thank you for the kind introduction, and thanks to the organizer for giving me the opportunity. The topic of today's talk is targeting using omics, novel interdisciplinary strategies adopted by our lab to combat drug-resistant microorganisms. So to start with, what is antibiotic resistance? It is a global problem. It occurs when a bacteria change, so antibiotic no longer work in people who need them to treat infection and is now a major threat to public health. And I'm sure that now all of you understand what a microorganism could do, because we are sitting now in COVID-19 era, and the reason we are not arranging physical meetings, we are into webinar, is showing how effective a tiny organism could do. And when we are talking about antibiotic resistance, it is about the bacteria which are not in few number, they are in millions. So you could understand the seriousness of the problem. And again, the seriousness of the problem could be again understood by what who is saying. They told that the threat of antibiotic resistance is no longer a prediction for the future. It is happening right now in every region of the world and has the potential to affect anyone of any age in any country. Antimicrobial resistance, or AMR, is the ability of the microorganism to stop an antibiotic from working against it. As a result, standard treatments become ineffective, infections persist, and may spread to others.
Hello, Dr. Hajra.
Yes?
Yeah, you can share your screen.
Okay, I did.
Visible.
Is it visible now?
Presentation. So kindly open the presentation, sir. Kindly open the presentation. Yes, sir. Yes, sir.
Out
So
and
please, yes, sir. Yes, sir. Please make it in slides of view. Yes, sir. Now fine.
Dr. Hajra, please.
Is it okay now?
Yeah.
Okay, sorry. So again, I was talking about antibiotics. As I told, antibiotic resistance is a global problem, and you all know about that because we are all sitting in the COVID era. The emergence of antibiotic resistance against popular antibiotics significantly reduces our arsenal to fight against deadly pathogen. And as WHO, World Health Organization, have stated, threat of antibiotic resistance is no longer a prediction for the future. It is happening right now in every region of the world and has the potential to affect anyone of any age in any country. AMR, or antimicrobial resistance, is the ability of the microorganism to stop an antimicrobial, that is an antibiotic, from working against it. As a result, standard treatments become ineffective, infections persist, and may spread to others. Misuse and overuse of antimicrobials is accelerating the process of resistance, especially in underdeveloped or developing countries like ours. So in our country and other developing countries, antibiotics could be bought directly in the sales counter without any prescription, which means there is no rule, there is no control to consume antibiotic, and people could consume antibiotic when they want it. Majority of antibiotics are overused and misused in animal forms. We all think about antimicrobial resistance, antibiotic resistance as a phenomenon which occurs due to the misuse of human. We forget that underdeveloped or developing country like ours, where most of the people are below poverty level, they could not even buy a regulated medicine for them. How they will buy regulated medicine for the animals? But when very costly animals got sick, they buy whatever possible, but they never complete the dose because of they don't have enough money. So the course is not complete. That results in existence of some of the bacteria which are resistant to those antibiotics, and that creates antimicrobial resistance. If you look at the effect, especially in livestock, that means animals, antibiotic consumption in livestock estimated 63,000 tons in 2010, which was almost double after 3 years in 2013, 131,000 tons globally. China, the United States, Brazil, and India remain the largest consumer of antibiotics in food animal. And the next sentence I'm going to talk about is good enough to say the seriousness. One child dies every 9 minutes from an infection caused by antibiotic resistant bacteria only in India. That's the seriousness of the matter. Also, over 27 million people get antibiotics unnecessarily. This is a beautiful picture. Here I have listed the invented antibiotics. The invention years are in green noted. The red notations are the year where actually resistance brought. And I'm not wrong when I actually indicated that penicillin, the magic bullet, was invented in 1943, whereas scientists have identified resistance in 1940, which is before the antibiotic even come into the market. That's what, the effect could be dangerous. So most of the potent antibiotics are showing resistance, reducing our arsenal towards combating against deadly pathogens. After 2011, that is very, very significant. After 2011, no significant antibiotics appeared in the market. There is always a war going on. We are bringing antibiotics and bacteria is coming out with resistance. But after 2011, a lot of resistance emerged, whereas we are not being able to bring potent antibiotic to combat. So kind of we are gradually losing the war. So we have to be very concerned. We have to invest our research focus much more here without thinking about the benefits, so that we could combat against, we could keep the existence of human community. So why drug resistance is such a big problem? It is a war. It is a war between human and microorganism, but that does not talk about the seriousness. And as experienced people know, this is very dangerous, because this is a war between husband and wife. That's what my experience says. And what I mean here, that this is a one-sided battle. So antibiotic resistance is considered as a one-sided battle, because if you look at the war, you clearly understand the wife is empowered with more harsher instrument. She is going to attack, whereas the role of the husband is just to defend. And before the war even announced, we clearly know who is the winner. So why drug resistance is such a big problem? Why we compare it with a problem of husband and wife? It's because extreme adaptability of the bacteria. Bacteria could exist in anywhere and everywhere. Bacteria could be living in very high temperature, in very cold temperature, in acidic condition, in basic condition, in very high metal toxic condition. That's the adaptability of bacteria. Amazingly small doubling time. If you consider E. coli, the doubling time is considered as 20 minutes, but actually they have overlapping doubling time that reduces the doubling time to even 9 minutes. So each 9 minutes, one E. coli is converted to 2. That's amazingly small doubling time, makes adaptability again much, favorable towards bacteria. Error-prone replication. Our characters are determined by our DNA, and bacteria is fortunate to have error-prone replication. This error-proneness leads to error, and sometimes that error actually becoming blessings to them. And nature, actually I'm coming into that, nature is using that error to help bacteria overcome the effect of those antibiotics and develop resistance. The last but not the least is a gene transfer mechanism. There are many theories about gene transfer mechanism, how it occurs through transformation, conjugation, transduction. But you know, to the audience, I could say in one word how simply and how dangerous this gene transfer mechanism is. This is a computer science department, so I'm sure that among the audience there are many people who are efficient in computer science. Now my research, I do experiments, but we also do interdisciplinary research. I'm a chemist in background, so I'm good about the molecular interaction, I'm good about the understanding the experiments, but I'm probably not that good to understand the computational processes. So it would be amazing if someone among you could volunteer and transfer your gene where the knowledge of your computer science is stored. You all are laughing, right? It is not possible, right? You cannot transfer your genetic character to me. Very surprisingly, bacteria could do that. Because of that, one tiny and harmless E. coli, if that become antibiotic resistance, it could have given that to Mycobacterium tuberculosis, Bacillus anthracis, all those potentially harmful pathogens. And here is the trick. Here is the game goes in extreme favor to them. But is that all? Is this adaptability, doubling time, error-prone replication, and gene transfer mechanism makes bacteria so superior in this game? No. To explain what I mean, I want to talk about, and first I want to remind you the story of Mahabharata at the beginning of the war of Kurukshetra. You all remember in the war of Kurukshetra, Duryodhana and Arjun both wanted the help of Sri Krishna. And Sri Krishna was clearly in favor of Arjun. So when Duryodhana and Arjun both came, and somehow Duryodhana came earlier, the condition was in favor to Duryodhana, and Sri Krishna, who is considered as God, even he was mentally partial, he could not show his partiality. So he made a trick. He offered his Narayani Sena in one side, and in other side, he announced that he is not going to take part in the war actively, but he would be there present. Now Duryodhana, being greedy in nature, thought, what I will do with the Sri Krishna who is not actually taking part in the war? Rather take the Narayani Sena, who are famous for their conquerness, who are like really aggressive and really, like a good support. So Duryodhana took Narayani Sena, Arjun got Sri Krishna himself, and rest is history. Why I talk about this story? Because I think in the war between human and microorganism, Mother Nature played the role of Krishna. We unfortunate human beings are the Duryodhana, and bacteria is the Arjun. And here, unfortunately, the human community even not given the chance. The choice of in which part Mother Nature, to whom she will favor, is actually decided by her. Mother Nature actually is thinking that, my analysis tells it, that this human community, I have given them so much power, they are empowered with intellect, they are actually ruling the world. But now, empowered with so much things, now they are developing antibiotics and killing the microorganism, the bacteria, the tiny bacteria who are actually my kid. So the philosophy behind the AMR emergence, human, us, develop the antibiotics to kill bacteria. But bacteria is a kid owned by the Mother Nature like human being. So Mother Nature wants to protect them. And ultimately, the war between human and bacteria transform into war between human and Mother Nature. And because Mother Nature, we all know, is almighty, and human is the strongest creature of Mother Nature, this is a very intense war and probably a never-ending one. So how could we win the war? Our strategy. If you ask me about the war, keeping my wife on the other side, I would not hesitate for a second. I would kneel down, offer the flower, and say sorry. In a quick manner, in a quicker manner, or in a quickest manner. Because my experience is, later or more delay I made, the flower would be demanded towards a diamond. So if it would be actually the relation of husband and wife told, there is no way we would have think about out of the box. It's just the way to say sorry. But fortunately, we are not fighting against our wives. So we have a chance. Our strategy for developing new antimicrobials looks like where we are being more specific, because they develop resistance. So we have to be developing strategies so that our design drugs, so if you would be a drug designer in 1960s, your goal for designing an antibiotic is to develop a potential small molecule which inhibit an enzyme. But being into 2020, your target is actually that, but as well as get knowledge so that the small molecule would be also less prone to the resistant mechanism. That's where the key of our discovery. And for these, we are taking the approach of omics, bioinformatics, genomics, proteomics, as well as metabolomics. To do that, we have to understand enzyme. We all know that biological enzymes are catalysts, but they are more than catalysts. They develop a magic pocket, a stabilized transition state, which is a key towards the drug designing. I'm coming into that. They expel water to give a very isolated, secluded, and unique three-dimensional environment for the reaction to happen. They bring reactive groups to start the reaction or speed up the reaction. And they take the help of enzymes. And as I told, it is not the substrate or the product. It is the transition state which have an intrinsic nature of binding, and understanding that is very important. But before going into that, before talking into that, let me tell you about an enzyme called beta-lactamase, because that is the focus of Hajra Lab. Before going to talk about beta-lactamase, you have to know about the story of penicillin. I believe many of us in our childhood heard about that. Alexander Fleming is a doctor who is working in a hospital of UK, actually Ireland, and he was working on Staphylococcus aureus. If you look at the slide, this is a device, or I would say plate, where bacterial culture is made. It's called agar media, where the small colonies are of bacteria, and this is the penicillium mold. So he was working in Staphylococcus aureus. He went for a vacation. He came back and he saw that in the plate there is a penicillium colony grown, and where the penicillium colony grown, the bacteria in that place actually died. Okay, or you better say inhibited. I would like to say in his own language, because that is exciting. When I woke up just after dawn on September 28, 1928, I certainly didn't plan to revolutionize all medicine by discovering the world's first antibiotic or bacteria killer. But I guess that was exactly what I did. So before going into the effect of the invention, let's look at the mechanism. Bacteria is a unicellular organism, so they try to make very strong end layer, which is called cell membrane or cell wall. So they made them by peptidoglycan layer, where N-acetyl glycosamine and N-acetylmuramic acid actually form those layers. These layers are cross-linked by a pentapeptide, L-alanine, D-glutamate, L-lysine, glycine, and D-alanine. And this cross-linking is taken care by an enzyme called transpeptidase. Interestingly, the DLA moiety is chemically very, very similar to penicillin. In addition, in penicillin, there is a 4-membered ring. And if you know thermostability calculation, you could easily find out that 4-membered ring, because of the presence of 90-degree angle, are very strenuous, so very, very unstable. So when this penicillin goes inside the active site, because of its similarity, the serine amino acid of this transpeptidase attack this carbon, break the ring, and form a covalent bond. Because of that, the transpeptidase become ineffective, so they could not take care of this cross-linking, and bacteria actually. So bacteria need the enzyme transpeptidase to make their cell wall rigid and cross-linked. Penicillin takes control of transpeptidase, they make transpeptidase ineffective. Bacterial cell wall are not cross-linked now, and the contents of the bacterial cell cannot be held in. Cytoplasm spills out and the bacteria die. So this is the mechanism of how penicillin or penicillin analogs, derivatives, which are called beta-lactams, are because of the presence of this beta-lactam ring, are working as a magic bullet. How penicillin changed the world history? In 1945, Fleming, Flore, and Chen were awarded the Nobel Prize in Physiology. Fleming, why you understand, Flore and Chen are the innovator of the molecular structure of this beta-lactam ring. And from 1928, when Alexander Fleming have invented penicillin, and to 1945, it is 18 years. And Alexander Fleming did not do further research on this topic. Think about, if you would be the first innovator of antibiotic, you would get funding at your ease. Then why you are not working for 18 years on that topic? He have replied in the Nobel Prize winning speech. He told that the overuse of penicillin might lead to bacterial resistance. Up to that, the world have not listened about the resistance. This is the first time the world is listening to bacterial resistance. But already penicillin have made a big impact. Why? It have a beautiful timing. In the First World War, before penicillin was invented, there was 18% death from wound-related pneumonia. People go to war, they didn't die in the war. They were wounded, and even they were slightly wounded, they actually got infected, and that infection causes pneumonia, and they died. In the Second World War, where the war intensity was much fiercer, wounds were much more invasive, less than 1% people died courtesy penicillin. Because of this one moment, penicillin became the hero. So beta-lactam was considered as new age alchemist's dream. Several new beta-lactam chemistry was invented and explored. And today, we all talk about money coming from pharma industry. Billions and trillions of dollars are flowing through many companies. If you probably heard about Merck, SmithKline, Danipon, Sumitomo Pharma, Novartis, all of them actually coming in the market because of one derivative of these beta-lactams. That was the effect of, beta-lactam. So there was money, there was fame, so nobody listened to Fleming. Also, I am showing you a Nature letter to editor, which actually shows in 1940, innovators from Oxford got idea about the existence of a penicillin breaking bacteria, but they didn't talk much about that because there was political influence, there was economy influence, there were influence from big market goons and all these things. As a result, beta-lactamase emerged. There are 4 classes of beta-lactamase. Class A, C, and D work mostly similarly. They work through the serine residue I'll talk about. Class B beta-lactamase, if you look at these dots, there are 2 genes. So they work through the metals. Coming to the catalytic mechanism, this is the beta-lactam drug. This is the protein part, the enzyme part. The serine have a hydroxyl group. The hydrogen is taken by the proton of the nitrogen and oxygen become O negative. This O negative attacks the positive carbon and form a covalent bond through a reaction called acylation. Because covalent bond formation is really a rare phenomenon in biology, so this enzyme transpeptidase become inactivated because it's stuck with this ring broken compound. Beta-lactamase is a evoluted transpeptidase. You understand what I mean? This is very key. Beta-lactamase is a evoluted transpeptidase which actually comes through a second round of reaction by which it could be able to throw the ring broken compound, so the enzyme is again ready to attack a second molecule, and that's what beta-lactamase enzyme is. Our goal is to understand this deacylation step with very high, in-depth understanding, develop small molecules which could inhibit this step, stop, or at least slow down. And I talked about like targeting ohm using omics. I talked about omics. What is ohm? Ohm is the intrinsic property, is to understand the transition state so that you could understand the enzyme properly and design drug based on that, so that that drug hit the intrinsic property of an enzyme. What is an intrinsic property? Suppose you are standing second in the class and you want to, stop the first boy or girl from being first, and you want to take a bad route. Okay, what you want to, involve him or her to watch movies, to, go to play cricket or play anything, and that's why he or she don't get time and you get time. So you will try many things. But if you know actually what that guy know, what the topper is actually interested, is he a movie buff? Is he or she is mad of cricket? If you know that fact, then you could have easily do what you want to do. That's called intrinsic property. Intrinsic property means the unique property without which the person or the enzyme could not work. My strategy is to understand these unique properties of the enzyme and utilize them towards the development of the drug. So our strategic planning involves interdisciplinary work, computational modeling, database analysis, virtual screening, simulation, understanding the loop dynamics, effect of point mutation, determination of binding energy, protein interaction, using them towards cloning and expression of the protein, doing site-directed mutagenesis, do protein purification, steady state kinetics, small molecule screening, biophysical, biochemical studies, crystallography, and develop a lead and ask our collaborators for chemical synthesis and this make a cycle to continue. That's what Hazra Lab does. And the goal towards this mission, we take DTMS approach. DTMS is diagnostics, therapeutics, mapping, and surveillance. Diagnostics, you all know, to develop some devices which could identify where new drug resistance is happening. So we have a device to detect. Therapeutics is design and develop new generation of drug. Mapping is measuring or recording data of a geographical location, let's say India, that is called mapping. And surveillance, once you map, you develop devices which could continue to see how that trend is going on. Is it enhancing? Is it decreasing? All this is called surveillance. Diagnostics, we have already developed a low-cost antibiotic susceptibility testing kit towards developing personal medicine. I have given the idea how to develop the construct. My friend Dr. Somadipto Mula from Berk, he have synthesized the molecule. Professor Nigel Slater from Cambridge University have developed a gel and cell mixture. Professor Monish Tiwari from University College London have converted this construct to a microfluidic chip so that it could be used in any places. And then lastly, the Richard Bauman, he developed a small microscope which could be bearable and very easy to detect. You put the chip there, according to the reaction, it could show a green light or red light. If it is a green light, it is susceptible, or it is a red light, it is resistant. Our concept is actually acknowledged by Royal Academy of Engineering as a innovation for the future, and we got Human Frontier Bioscience Award for that. The next one is BL tester or beta-lactamase tester, which is an antimicrobial outdoor test kit. This is based on a compound called nitrocephin, which is a beta-lactam compound. So it is affected very uniquely by beta-lactamase enzyme. And when there is a presence of a beta-lactamase enzyme, it actually changes from its yellow color to a very bright red color. My PhD student Nitish Pandey have worked on this. He have developed a 3-component system BL tester, which actually help any sample, especially out of the outer field, without doing microbial culture. So it is very sensitive and very easy to use. So simple but highly sensitive colorimetric test kit. BL tester could work on environmental samples. It may work on crude sample, for example, soil, wastewater, urine, anything, vermicompost. BL tester could work on any animal form for surveillance and drug resistance monitoring. So this is already done and patented. Now we are working on BL tester basic to next part, which is called BL tester advanced. In the first part, this is Dr. Sandeepan Haldar from VNIT Nagpur. He, along with us, we have developed nitrocephin derivatives, which are more intense color producing compounds. He is currently synthesizing them, as well as he is also synthesizing something called siderophore. This could allow us to detect efflux pump mediated drug resistance. What I'm talking about, there are some resistance where you put the drug and there is a pump which will throw the drug out. But if you have siderophore, they make a hole so the drug could take entry even if the efflux pump is present. So these two compounds actually help us towards developing the newer part. They are currently synthesizing 100 such combination of compounds, and we are going to test them. In the part 2, we are going even towards more switching. Again, we are taking the help of Manish Tiwari from University College London, who actually did PhD with me, a very good friend of mine. He worked on mechanical engineering and he's working on nanofluidics. So with his help, we are developing various type of microfluidics chips where we could detect is it penicillin resistance or cephalosporin resistance or trimethoprim resistance, ciprofloxacin resistance, kanamycin resistance, different type of resistance. So the switches we could understand. I don't like, we are working a lot on that. I would not go into detail, but because of patent issues, but with Professor Narayan Mishra from IIT Roorkee and Arup Samanta again from Physics Department IIT Roorkee, Professor Mishra is working with design, synthesis and testing of enzyme nanoparticle corona towards diagnostic development, and Arup is helping me towards enzyme quantum dot interaction based imaging for further detection of microorganisms. So these are, some of the example I'm showing you where we are working towards development of diagnostics, because diagnostics is an extremely important criteria for antibiotic resistance. Then I talked about therapeutics. We have done many, designing of many compounds, and we are still designing. We designed boronettes. We designed mechanism-based inhibitors. We design different type of carbapenems, as well as we also test them. We design a compound which is called NXL104, that is actually a DBO moiety compound. We are still working for better moieties of that, and we also work on novel penems. So there are different class of compounds, and we are constantly designing and asking the collaborator chemists to come up with several such compounds. Something I want to talk about here, we are now, ours is a young lab, but we are equipped with instruments needed for all this in vitro testing. By in vitro testing, I mean we could test the effect of drug on a target enzyme in their pure form. We are also having entire set of ex vivo testing. By ex vivo testing, we mean the testing with the real bacterial system. We also have collaborators who are helping us to do cell-based testing and testing in animal models. We could also do it by ourselves, but as you could understand, our labs are actually full of bacteria, so we don't want to bring eukaryotic, testing in our lab. We also have few unique setups which gives us very interesting type of testing. I'm going to talk about them in brief. We have loads of newly designed small molecules through our studies. We are open to collaboration if someone wants to synthesize them. We are also open to any other form of antimicrobial setups like nanoparticles, nanoclusters, MOF, as Dr. Koyal was talking about, natural products, all of them you could, be free to discuss with us. We could test them properly based on wild type bacteria as well as drug resistance bacteria. All are actually there and well characterized in our lab. So as I told, I want to talk about some unique strategies, develop and analyze database to set up our targets, how we are doing it, combining computational and experimental technique, and design novel transition state analogs and mechanism based drugs. I talked about few of them, but I'm going to talk about the mechanisms and methodologies which favors understanding in-depth catalytic mechanism. So how we form the database, how we do the filtration, rename and processing, all the process I'm coming into. But before that, as these are something, if I don't go into basic, you don't get what we are actually doing. I want to talk about some basic, information regarding decoding biological macromolecule, which actually make a revolution in biology. It starts with human genome sequencing. What is the concept? If you look at the DNA have 3 component: nitrogenous base, sugar, and phosphate group. Among them, the change only is there in the nitrogenous base. There are 4 nitrogenous base which are called adenine, guanine, cytosine, and thymine. Now, if you look at them, they are actually extended in a DNA synthesis by using this 3' hydroxyl. Now, if you take out this 3' hydroxyl, they are DNTPs, they are called D-DNTPs. What is the interesting part of designing or taking this hydroxyl group? If you take this hydroxyl group, they would not help elongating the DNA. So in a DNA synthesis, it would stop once a ddNTP is incorporated. And that is used in a sequencing technique called Sanger sequencing technique. So what is the technique? Before that, you have to know what is called PCR, polymeric chain reaction. Polymeric chain reaction is a device setup done by Carey Mullins, who got Nobel Prize for that. What he did, if you have DNTPs, okay, you add the enzyme, you add the buffers, and you run the cycle, you get one. If you have one copy of DNA, it would be 1 to 2 to 4, 8, 16, 32, 64, 128, and it goes like that. So after certain cycle, any tiny amount of DNA would be enhanced to a big amount, and you easily detect that DNA. Carey Mullins' PCR innovation have revolutionized the biology, but it is further revolutionized by Sanger utilization. What Sanger did? Sanger have set up that, PCR reaction, but instead of DNTPs, he also add ddNTPs. So instead of getting one big fragment, now wherever the ddNTPs is entering as a competition, it's the DNA elongation stopped, and you get all those fragments. Okay, those ddNTPs later actually labeled with fluorescence, and instead of separating them in a gel, they are separated with a capillary electrophoresis, so the sensitivity of their differences are more pronounced. Now laser is thrown to the capillary, and because there are fluorescence like probes, they have different effect, and from the different effect their data was read out, and from there you could read the DNA. That's the first time we start reading a biological macromolecule in a automated manner. That's what the revolution. But that's not the complete revolution, because in Sanger sequencing, you could only sequence 800 plus nucleotide base pair, but the good part is they're very accurate and they are considered as gold standard. Now, Sanger sequencing was revolutionary because you have now you get the gene, and gene is related to mRNA. So you get the sequence of mRNA. mRNA is related to protein with a 3-letter code of the nucleotide to a amino acid. So this is called codon. So if you know the sequence of a gene or DNA, you get the sequence of a mRNA, you get the sequence of a protein. So now you are getting into reading all the biological macromolecules which are actually talking about the character. Okay, so but the problem was you could read the DNA, but now you want to read the entire DNA. The whole DNA content of the cell is called genome. The whole mRNA content of the cell is called transcriptome. The whole protein content of the cell is called proteome, whereas the molecules generated through the action of the enzyme is called metabolome. So if you could read the genome, now you could read the gene, but if you could read the genome, you could read the transcriptome, you could read the proteome, and you could read the metabolome. That's where the revolution. Now, why gene to genome was very difficult? Because a tiny virus is even have a size of 170,000 base pair. Remember, only 1,000 could be sequenced by Sanger, and this is 170,000. A bacteria having 4.6 billion. A fruit fly is 130 million. A human is 3.2 billion. And a canopy plant is 150 billion. So the genome is so big that I could give you an example, and you could get easily. If you could fold out, if you could open up the chromosome which is folded in your body, in one cell of your body, you could go to sun and come back 60 times. That's the length of the DNA. in a genome. So you need, so Sanger was first generation sequencing. You need next generation sequencing where you need to generate many million of short read in parallel. You need to speed up of sequencing process compared to the first generation. You need low cost because Sanger costing, you know, like 1,000, 2,000, and for one virus it is 117 to 2,000 rupees. You understand the like hugeness of vastness of the money and the automation. So that's all happened in the Human Genome Project. What is the effect of Human Genome Project? Before Human Genome Project, a scientist is working to identify a gene in his or her entire PhD. So if you look at upper panel is my first 4 years of work, but so like this is the small amount of data this poor PhD student have gathered through sequencing. This is before genome sequencing. And now after human genome sequencing, an angel is coming to the God, the human genome code is been unraveled. And God is also, you know, like afraid now. He said, damn hackers, now I have to change the password. That's the effect of, you know, Human Genome Project. It takes 13 years and it was considered as a small step for man towards going to heaven. That means it is a step towards immortality. It was published in all the very, very acclaimed, you know, popular journals as well as scientific journals. And it was so big event that the then President of America, George Bush, came personally and collected the entire data. Now you understand how big is that. And though this is 2003, we take like around another 12 years to be habituated with the vastness of the data. So this comes only in 2015. So in this talk, any young scientist, any young you know, researcher, any young student who are present, this is the future. This is where biology is now studying. In that, it's a level up. And what is the effect? See, like before, people have Department of Genetics and Biochemistry. People want to look at the effect of one gene. Now people are taking all the genes, looking at, like, some patient have cancer, take the like tissue from the cancer affected zone, take the tissue from the normal zone, do genome sequencing, compare them, get the result. So biology is understood in different level. Okay. And in that time, there was, you know, as I told, the coining of ohm word happened. One scientist was interviewed. How do you define success in science? And he says, when I could coin my own ohm word. And now you look at the cost. The cost starts with like more than 100 million, and it is coming into much, much less already in 2017, standing in 2020. With $500, you could get a complete information about your genome. So coming back, those genome data have given a lot of boost, a lot of information. I am here introducing my postdoc, Rajesh Gajara. He's an excellent postdoc. He had done his PhD from Brazil, and I'm fortunate that he came back to my lab and he is taking care of all the genomic related analysis. Whereas I again introduce Vivek Jungare, who is a PhD student. He is doing all the bioinformatic analysis, and when Rajesh is going to the genome level, the gene level, protein level is taken care by Vivek. So what they did, they have collected already developed like sequences from already developed, you know, databases. They did the unification of the sequences, which means removing identical or non-relevant sequences. Then they are going for critical incorporation of different organisms, different gene names, sequence length, etc. Then they change it to so that we could access. What I mean, like suppose the data is a gene data or a protein data or a structure data, they develop a rename as if they could call them together. Okay, then they perform analysis through multiple sequence analysis, phylogenetic analysis, and many more. So they develop a, like for class A beta lactamase, they develop a 12,000 protein from the database, and they use different genes, different organisms, excluded sequence with organism not specified, excluded sequence with higher divergence like partial sequence, and finally it comes down to 12, 16 sequences, and then they develop the phylogenetic tree, and they like arrange them according to the closeness of the phylogenetic tree coming on the criterias, and they are all a lot, and then they have classified, and to our surprise we see that these are coming into clusters which are actually like few of them are already being into standard grouping, and our research bring those already known of them and others which are not known. So based on them, we have already classified 93 subclasses of class A beta lactamases, and what we did, we are doing cloning, overexpression, and characterization so that we could work on those enzymes, and we decided to complete the whole thing within coming 4 to 5 years. Coming to, so that's our effect, combining our analysis, sequence based, genome based analysis with our like work which I'm coming, we have characterized each and individual classes. Now we combine computational and experimental techniques, where if you look at, we have done structure analysis, we have done alignment, we have gone through their differences in structure, we have studied the active site where the reaction happens, we have studied the omega loop, which is a very interesting loop present in class A beta lactamase. We go for SDN loop, we go for KTG motif, we are working for SXK motif, and all those things we did. And this is a study of class A beta lactamase simulation. This is done by another project student, Muskan Vambi, who is a BTech student but done amazing work in the lab. So these are the omega loop, and we had done their simulation. So before we are looking at the static data, now we convert from the static data towards dynamic and look at the difference in the level of dynamism. And then we come to the cloning and overexpression. Looking at the band, you could have understood that we have get the pure protein. Ghazal is a PhD student who have already graduated. She have helped making those constructs, structural constructs. So we take the gene and we make the construct in such a way so that they are structurally engineered. This is Subhecha, who is working on metallo-beta-lactamase. This is MSc student Swami. This is Abid and Kunal. Abid is working on, again, metallo-beta-lactamase and a new type of beta-lactamase coming from a class which is a positive beta-lactamase. We are talking about it. Kunal is working on class A carbapenemase, another PhD student. So I didn't talk here about Nitish, whom I talked about before, and about Sorjo, whom I am going to talk about next. So study of enzyme in solution, we had done kinetics, biophysical, biochemical methods, and some special techniques. I will quickly go through. This is the kinetic study where you will see that a colorless solution is converted to very intense red color, and based on the changes we actually measure the red, kcat, km, ki, and all those things. These are some of the example of class A enzyme: SHB, GES, KPC, BLAW1, F2, and oxi is another from another class. So we compare them with all the different class of beta lactam drugs and beta lactamase inhibitor to understand their potency. Biophysical studies, we have done circular dichroism, which is helping fixing one point. Now for a particular protein, you could measure anything. So if you look at, there are 3 points. You will see 1 peak, 2 peak, or 3rd peak. You could fix any one of the peak, take the point and then see the effect of temperature, pH, denaturants, anything, and that's what circular dichroism would very intensely measure. Then fluorescence spectroscopy, you all know about Zablonski diagram. Fluorescence spectroscopy, especially I'm talking now about class A beta lactamase, they have tryptophans which are intrinsic fluorophore, and we utilize that intrinsic tryptophan towards developing specific assay. We mutate the other ones which are not important, so that now we look at the binding of a drug based on the movement of the tryptophan. Differential scanning calorimetry or DSC, it is the thermal denaturation of the molecule associated with the heat change. It also measures the enthalpy associated with the unfolding of the protein. We use those as a technique. Then isothermal titration calorimetry or ITC is an amazing technique. In this technique, you could measure all the thermodynamic parameters of protein drug interaction, like enthalpy of the interaction, Gibbs free energy, binding affinity, and stoichiometry. Without this machine, there is no, available instrument where you could make stoichiometry. What is stoichiometry? Stoichiometry tells you if you have 1 mole of enzyme, what mole of small molecule, what is the ratio of small molecule binding to the enzyme. This is very, informative data towards understanding in-depth molecular interaction. Then microscale thermophoresis is a kind of unique instrument. It have 16 channels which are made of microfluidics, nanofluidics. So these are nano channels, and all of them would be filled with first one is only protein, and then protein and other drugs. They are all labeled. This is the labeling, this works through labeling technique. So the protein would be labeled with fluorescence, and then the heat would be coming through laser, and you could measure binding affinity KD on these. So these are techniques we commonly use, but there are some unique strategies Hazra Lab also take. Coming to the catalytic mechanism I talked about. So you remember the deacylation we are looking at. So we develop mass spectrometry based kinetic study where we are studying, different molecules. See here, we could identify how the molecules are breaking, and even we could identify if any novel fragment is coming. And mass spectrometry is very sensitive, so we do time-based mass spectrometry and get the kinetic data about that. So this is mass spectrometry with kinetics, which helps understanding the breaking of each and every fragments. So if you see, you could identify all the fragments here. And now coming to the process, which is mass spectrometry, we work on carbapenems. You could easily see that how the shifting happened. So this is the one when the carbapenem is not affected by the drug, but when it when it affected, there is a shifting, and you see that how long this shifting occurs. So this is the protein, and then the carbapenem comes and attached to the protein. If it comes out of the protein, then the peak would be returned here. So based on that, you could see the inhibitory effect of the drug by this technique. Another technique which is very unique is called Raman crystallography. Basically, this is a confocal microscopy setup, but we have included a video CCD here. So you could have seen the live reaction which is going on the focal tray here, where we put a crystal tray. The crystal is kept here, and this is a protein crystal, and we also put a Raman monochromator instead of a light. So the ray is coming is a Raman spectra, not a light. Okay. So when we put the crystal and we put a small molecule, the small molecule only react when it goes inside the crystal. In the solution, it could not react, and the focus is on the crystal, so you will only look at, so the confocal setup is looking at the 3D width of the, because confocal is looking at the z axis, so it could only catch the incidence happening inside the crystal. So if you have a small molecule, you put it inside, it goes inside the crystal. If it would change, there will be continuous change in the spectra. We will align those change like those spectra, and if there is a change, we would exactly find what is the change, when is the change, and by calculating, applying quantum mechanics, we could say what is the change of the functional molecule. So this is a setup by which you could actually see a live reaction happening inside the crystal. And this is an example where these three drugs, if you look at these three drugs, looks very similar, but we have shown the difference in their reacting time. We have also shown the effect of mutations happening here. So if you look at the spectra is here, and when the spectra are compared, you could easily find the changes, the width of the spectra, the timeline of the spectra. So this is giving a real in-depth knowledge about the reaction mechanism, which we want to know, because our work is to understand the reaction mechanism and find out the development of the transition state. So as I told, if you look at these three drugs are pretty similar. No other technique could have differentiated them, but by developing this Raman crystallography setup, which in IIT Roorkee we have developed, we have differentiated them. So coming to the summary, we have opened a Pandora box of one of the most interesting and demanding problem of current era, which is antimicrobial resistance, focusing on beta-lactam. Our study using interdisciplinary approach would help understanding the subtle differences between each and every beta-lactamases. As we have shown, we have designed a FRET-based switch for detection of normal and drug-resistant bacteria, as I talked about. This would be helpful towards going to personalized medicine. Those understanding, as I showed on diagnostics and therapeutics, would also be helpful in designing new generation theranostic, that is therapeutics and diagnostics, especially in testing newly invented drugs. Unique techniques adopted by us is helpful in understanding the catalytic intertalking, hence better understanding in reaction mechanism. Within another 4 to 5 years, we would be capable of developing an integrated platform for complete characterization of beta-lactam antibiotics. What do we mean? We are now characterizing each and every special beta-lactamases. Now, that would be integrated under one device, where any new company who are actually developing beta-lactam or beta-lactamase drug could be doing a holistic testing. It would not be a testing towards 1 or 2 gram positive, gram negative bacteria. It would be rather testing on 96 different beta-lactamase, and they have different properties, and all the properties are stored in the library given by us. So that's one of our major goals. Now I'm talking about understanding structural reason behind drug resistance, experimental validation of our theoretical hypothesis. So I told you we are looking at the natural variants, how they're changing, how the change could be understood, how we could understand what is nature doing. If we could understand what is nature doing, we could be always one step ahead to understand what is coming next. So before any bacteria or any virus, as we have seen, come out with a pandemic, if we have those understanding, we could be alerted about that. So these are, drug resistant variants. You go to hospitals, we have a lot of collaborators. They are giving us the clinical isolates. And today I would talk about one which is belonging to SDN loop. If you look at SDN loop, in all the organisms, beta-lactam is there SDN, but only in Mycobacterium tuberculosis and Mycobacterium bovis, they are SDG. So there is a glycine amino acid instead of asparagine. We wanted to check what is the effect. And what we know that PEM, which is an E. coli resistant variant, were found to contain serine to glycine mutation. That means the serine is actually mutated to glycine in many organisms. We found it in E. coli. It's reported in TEM76. It also reported in Klebsiella, Proteus, Shigella, and Citrobacter. Very interestingly, tuberculosis, we don't find this serine to glycine mutation. Before going into that, I want to tell you why this serine 130 is important. As I told you that serine 70 is the catalytic residue, so it do the first attack. Serine 130, especially for the mechanism-based drug like sulbactam, tazobactam, clavulanate, do a second attack and develop a cross-linked product. So the drug, most part of the drug is taken out and rest of the part is cross-linked the two amino acid of the enzyme serine 70 and serine 130, so the enzyme become really inactive. But if you could make serine 130 mutated, you could develop resistance towards this drug. Okay, clavulanate, sulbactam, and tazobactam. So to test why tuberculosis is not developing, first we compare their structures and we compare their secondary and tertiary structures, and we want to, before going into experiment, we wanted to do theoretical studies to save our money. So we wanted to do molecular dynamic simulation. Molecular dynamic simulation, as I told earlier, is an in silico method that mimics the in vitro environment in which a protein actually mediates all its normal biological functions. MD simulates the movement of all the particles in a molecular system by iteratively solving Newton's equation of motion. So if you think about a protein, it is connected, covalently and non-covalently. For covalent, it would have stretching, bending, and torsion. For non-covalent, it would have hydrogen bond, Coulombic interaction, and van der Waals. All those 6 interactions are put into a force field, and this force field was applied with Newtonian equation of motion to see how the enzyme is moving and what is the effect of the movement. So we developed MD simulation for Escherichia coli beta-lactamase and tuberculosis beta-lactamase at 300 Kelvin and done for 100 nanosecond time scale. It has been performed in GROMAX 4.5.4 version using AMBER 99 SB ILD and force field. So there are different programs: GROMAX, AMBER, LAMS, CHURM, and all those things. So we have used GROMAX here. And we use, when you are theoretically using, you have to use the water molecules as model. So we use TIP3P water model, and we do energy minimization using steepest descent algorithm for 50,000 steps. So you will see the effect when in E. coli, it is very stable. The SDN loop is very stable. In SDG loop, we have already, see the disturbance. So we thought that because of the presence of this glycine, or you could say the absence of this asparagine, it loses some interaction, so disturbance comes here. And if you see S130, like the serine and aspartate are having like much more fluctuation, much more disturbance in the movement than what in the E. coli. So we decided to do objective. We wanted to develop S130 mutant variant of tuberculosis. What I mean, as I told, S130 was mutated to glycine in E. coli. We want to make so tuberculosis by itself didn't do that. But what our thought was, tuberculosis being the king of pathogen, why it is not doing? So we understand the inner reason behind it. So we ourselves play the role of tuberculosis and mutate that. Okay. And we want to validate the hypothesis. Like if we mutate, can we get the drug resistance? And then we also want to like test the stability of the mutant, and if possible we want to solve the structure, because we are structural biologists. We wanted to see like whatever we see in theory, okay, but is it actually happening in experiment? So we want to solve the structure, crystal structure of the protein. So we clone and express the protein, and you see that we clearly see. So what we did, we mutated to glycine and alanine, and you could clearly see that whereas the wild type protein is produced in high amount, the productivity is clearly reduced in S130A and significantly reduced in S130G. Then we perform, reactions with nitrosapine, which the reporter molecule I talked about, and ampicillin, and you see that the reactivity is like the Km values enhance and kcat value significantly reduces. So the efficiency, which is kcat by Km of the enzyme, is reduced here. Okay. Also, we have seen that against those drugs, the mechanism-based drugs, clavulanate, sulbactam, and tazobactam, wild type have good activity for clavulanate, sulbactam, and tazobactam, whereas there is very high value required. That means resistance happened in case of the G and A mutants. Now, we also wanted to test, is it what we see in vitro? Is it happening in vivo? So what we did, we take the cells, and when we add MBI as a drug, we see that the drug actually kills the bacteria. Whereas when we have the mutated one, you see the bacteria is not killed. So in vivo, our data is actually validating what we get in vitro. Also, so this we did for E. coli, okay, gram-negative bacteria. For gram-positive bacteria, we see the same thing. When we have the wild type, the drug is actually killing, but when we have the mutated form of the bacteria, the three-dimensional structure of the bacteria is clearly maintained. Now we come to the analysis of the stability. So we did that. We take the spectral and CD spectra, and the CD spectra is clearly changed from wild type to the mutant. And when we did the temperature, to our surprise, we find that when the wild type shows a TM of 64 degrees centigrade, that is reduced to 49 for S130A mutant, but it is again further reduced in case of 130G. It's a nearly 25-degree reduction only for one mutant. As I told, we are crystallographers, so we do further purification using size exclusion chromatography, and then we solve the structure. So this is the complete structure. I don't go into the detail. I'm just showing you the difference. See, generally with one mutation, point mutation, we don't see movement in the protein structure, and definitely we don't see movement in the backbone, because protein backbones are very rigid. Here we see a movement of around 4.3 angstrom, which is a very, very, very significant move, I could say. So as I told, this entire work was done by Shaurya Bhattacharya. So in this work, we get SDN, which is a conserved loop in various organisms except for Mycobacterium tuberculosis, which has Gly 132 in replacement for conserved asparagine 132. Asparagine 132 to glycine replacement provides Mycobacterium tuberculosis with higher flexibility of the SDN loop, as it can be concluded from the comparative RMSD and RMSF analysis of the SDN and SDG loop. The smaller TM of the mutant protein support the fact that when mutation of S130A and G in Mycobacterium tuberculosis confer the enzyme instability, I want to enhance this. That means probably like E. coli or Proteus or Klebsiella, tuberculosis also try to generate this mutant. Nature is pushing tuberculosis to make this mutant, but because of the asparagine to glycine mutation, this mutation cannot take place because the enzyme lose its stability. Structural study comes up as a surprise, as I told, validating nature's diversity at the end. And this is a good example of interdisciplinary study towards solving problem of antimicrobial resistance problem. So coming to the conclusion, emergence of antibiotic resistance is a natural process. We could not stop it, but we could definitely slow it down, and that's what our goal here. But what is terrifying is the trend in developing countries. In India, we had 29% MRSA, which is drug-resistant Staphylococcus aureus, detected in 2008, and in 2014 it is enhanced to 47%. It is not the scientists or the doctors, it is all of us who have to be together in this mission. As you already know with COVID-19, how a tiny organism could do. So we all have to be aware about it. We all have to be, coming into a mission. Like a lot of companies, they don't come and encourage our projects because they know that the drugs we are designing, even they are much better, much intelligent, much smarter. But because nature is against us, resistance is going to happen. And that's why we don't get, funding from the companies, because they are professionals. But the country leaders have to understand that, and we have to make a significant change also in our lifestyle, have to demonstrate others about those good life practices. So students who are here, please spread the message about the drug resistance to your friends who don't understand about that. You have to convince the entire world about the seriousness of this, and that's how we could combat. And from Hajra Lab have adopted an interdisciplinary combinatorial approach I am talking about to understand the holistic mechanism of antibacterial resistance and working relentlessly towards combating against this. But to be very honest, to be successful, it needs many other experts to join us in this. So in this platform, I am asking people to come forward, understand this problem, the importance of the problem, and join us in the war, because losing this war means questioning the existence of human community. That's serious it is. Without antibiotic, there would be no pregnancy, there would be no operation, and our human life would be devastated. With that, I would love to thank the funding agencies who actually help us running. We got funded from CERB, we got multiple fund from ICMR, we got funded from DBT, we got Spark in the UK, we got from Department of Health Research. Recently, we got UCOST, which is our Uttarakhand. We are also funded from Royal Academy of Engineering, UK. And last but not the least, IIT Roorkee, the administration have helped us with many, financial helps, helping us buying instruments which we don't get from funding agencies. Recently, we got the protein X-ray crystallography machine, which is a good addition to our work. And again, about Hajra Lab, I have no words to express my thankfulness to the team. We have a team of 30 people, but in addition to that, another 60 people every year come and work with us to develop databases, to work on, different bioinformatics projects, and I am thankful to everyone. With that, I thank the organizer for giving me the opportunity. I thank the audience for listening to me, and I'm open to question. Thank you very much.
Thank you, sir. Sujay, sir.
Switch on
Thank
your
you.
video. Hello, Dr. Hajra.
Yes.
Yeah, please switch on your videos. Hello.
Hi.
Actually, we are running out of time.
Yeah.
Yeah. Yeah, yeah. Okay. Thank you, Dr. Hazra.
Thank you,
Very
sir. Thank you, sir.
fabulous and fantastic presentation. Okay.
Thank you.
Actually, this is my opportunity, our opportunity to, actually, we want to go your Hazra lab in future.
Sure,
If you
sure, sure.
Yes.
All are welcome.
if you
Yeah,
give
yeah.
us the permission. Okay.
Sure. No, no, no. Don't say as
And
I told. This
Yeah,
is a
yeah.
type
not
of
only,
work we can only do having a big group. This could
Yeah.
not be an individual lab's work. I am
Actually,
fortunate to get a lot of people. I have 3 postdoc and 13 PhD currently, and a lot of MTech, MSc, BTech students.
not only us.
But still, it's not one lab's work. It should be that we should have a big Indian community to work on AMR, and we are actually trying to develop an organization called Antimicrobial Resistance Alliance, where a lot of veterinary doctors, um, clinicians, they are coming. So we try to make a platform of clinicians, veterinarians, and scientists into one platform.
okay. Madhumita madam, are you here? Have you any questions? Hello, Madhumita madam. Hello, Madhumita madam, are you there?
Yes, yes. Madhumita ma'am, please unmute your mic, and if you have any question, please ask. Madhumita ma'am? Madhumita
Okay.
has her mic.
So now, Dr. Shuja Mistry, please ask me questions and vote of thanks for the and conclude the session.
So, if any participants have any questions, I think actually we are running out of time. Another meeting is actually done in this platform maybe from 8:30. So all the staffs are actually asking for as we finished this webinar. Actually, as myself and Dr. Pudut Sarkar think about these topics, we know that this is not possible for within this limited time period. We can cover this huge field of this type of field.
Right.
So
Also, for myself, I introduce basic slides because I
yes,
generally
sir.
talk in biotechnology departments and all, so they know about what is gene sequencing and all. But this is a computer science department, so if I don't talk about them, it just doesn't make any sense.
Sure, sure, sir. Actually, our target is that one part is that is science, that is basic science, that means that's bioscience and things, everything, and another part is that computational part, and that is very important. That now the collaboration of these two fields, computation and the bioscience and the science of. So it is very important for us. Basically, not only us, our student of this university and other university also wants to visit your labs and other labs, as they can enrich this field. Actually, they know this field, and so this is one of our motto. And that's why we want to do a workshop in future. Hope all our distinguished eminent speakers come to our physically and deliver their eminent talks, and that can they give their knowledge, whatever they have share their knowledge to all the students, and our student will be benefited. That is our main goal actually. So with this, I just what I said, that it is my great honor to propose the vote of thanks to all who have helped us in making the seminar such a resounding success. Actually, myself, by Dr. Sujay Mistry and Dr. Poddu Sarkar, on behalf of the Department of Computer Science and Engineering, and as the coordinators of this event, extend a very hearty vote of thanks to Honorable Vice Chancellor for giving us the opportunity to organize this event, and also for gracing today's seminar and delivering the inaugural address. Thank you for your interesting and thoughtful, provoking address. I would like to thank our distinguished speakers, Dr. Subhashish Barik, Dr. Shogat Hajra, Dr. Ahmed Ibrahim Samir Khalil, and Dr. Shivnath Koyal for making excellent presentation and making this webinar interesting and meaningful. Thank you all for sharing your specialized knowledge with all of us. I would like to thank our head of the department of CSE, Dr. Suparna Bishas, and Director of School of Computing, Professor Devashish Dev, for their unconditional moral support and guidance. And now, I am happy to express my vote of thanks to all our staffs members working tremendously behind the scene. Especially, I would like to mention the name Mr. Prem Kumar for his all-out effort. And without his technical support, it would be not possible to us to make this webinar a grand success. With this words, finally, this event wouldn't have been successful if such a great number of participants will not come out. I would like to thank all the participants from several institutions and from abroad also for their active participation of this event. Thank you so much for your encouragement, presence, and cooperation. Hope taking feedback from your side, and as per your demand and suggestion, we will soon organize a workshop where we will discuss all these topics more detail with these eminent speakers. Once again, thank you all. Thank you for your cordial cooperation. Thank you. Thank you for all.
Thank you.
Both the two.
Okay, good night to all of you, and thank you all the speakers for participating in this webinar. Thank you, thank you, thank you. So good night.
Thank you, everyone.
Yeah.
Good night.
Good night.
Good night. Good night.
Good night.
Thank